was at Bombay. And
good. So now it's very convenient through the internet, through Zoom, but it is not the same experience.
Nice seeing you, Thomas.
Likewise. How are you doing?
Yeah. Good, Thomas. Yeah, thanks for speaking at our conference as well last year and promoting us.
Yeah, no, it's good initiatives. I'm glad we can help.
Thank you.
So can we begin, Dr. Patil?
Yes, sir, we can begin now.
So warm welcome to everybody. It's 7 o'clock, real coffee time in India. So 7 p.m. rather. And we had a very good session in morning. The conference was formally launched, and we have presentation from speakers from India and abroad. So now we have another very good friend of our society, and also a friend of alternatives, and promoting the cause of alternatives in a very passionate way. So we thank you, Dr. Thomas, for accepting our invitation and to be part of this kind of movement we have launched in India about alternative animal experiments and how we can go ahead for various aspects of its implementation and validation of the methods. In the afternoon session, we have a lecture from Dr. Pant and other speakers. They spoke a lot of latest developments which are happening in India. So we are very grateful to you that you accepted for today's invitation. So let me introduce for the benefit of the audience and young audience. Many of them are there, over 100. And the event is also being streamed live on the YouTube platform. So I think many people have joined it, and there was almost no registration fee, and many young researchers are with us. So let us introduce Thomas, who is a PhD and also is a chair for the evidence-based toxicology in the Department of Environmental Health and Engineering at Johns Hopkins Bloomberg School of Public Health, Baltimore. He has a joint appointment with Whiting School of Engineering. He also holds joint appointment for molecular microbiology and immunology at the Bloomberg, at the Georgetown University, Washington, D.C., and also holds a joint appointment as a professor of pharmacology and toxicology at University of Kansas, Germany. So he's a director. He's the director of the Center for Alternatives to Animal Testing, very popularly called CAAT, of both universities. And CAT hosts the Secretariat of Evidence-Based Toxicology collaboration, the Good Red Cross practice collaboration, the Good Cell Culture practice and collaboration, the Green Toxicology collaboration, and the Industry Refinement Working Group. And he, as an investigator, he heads the Human Taxome Project, very interesting one funded by NIH Transformative Research Grant. He is chief editor of Frontier in Artificial Intelligence. He is also the former head of the European Commission for Center for Alternative Validation, very popularly, and one of the renowned and one of the rather first validation center in Spara, Italy, IBCAM, and has authored many papers, more than 500, and H-index, which makes us envious. 99. Have you crossed 100, sir? H-index, something very great. So
actually, we just passed 100 last week. So.
Oh. So we must congratulate you. Very rare feat. Generally, we published about 300-400 papers publication. Then also so it means you are well led, well cited expert, and we are very pleased that you spare your time in maybe very chilly morning outside. So we like to hear from you on the different aspects of this alternatives. And I think you will be talking about good cell culture practices, I guess, even where also cell cultures are also a biological material. So that should also not be wasted. So like for animals should not be wasted. There should be refinement and reduction in the animal. So why we unnecessarily waste animals cells also, which are so precious in the time of this stem cell development and also in the gene technology. So I think, Thomas, we are very keen, something very unique type of lecture it will be for the young as well as experienced toxicologists and biologists and different field people. So Thomas, I again welcome you to this forum. And I would like to just request you to kindly go ahead with your presentation if you have. And now floor is yours, sir.
Thank you very much for this nice introduction. I'm very grateful to join my Indian friends and some international ones here in this meeting. It is always a pleasure to be with you. And I have done a very nice introduction already to my talk. I want to focus today on the quality of cell culture, because that's something which I think is the only way we can really make them substitute for animal testing by being of utmost quality in what we are doing. I make my slides available for who is ever interested. I have met a QR code here. Don't worry if you're not fast enough with your camera, I will show it at the very end of my presentation again. I also have quite a few QR codes on for any papers you might be interested in following up. I deposited them in this work. Okay, before getting started, really, I would just acknowledge that we are working differently at the moment, working online. But this has also some good sides. It was very easy for me to accept this invitation without 2 days of travel. But also we see an enormous uptake of all of the international audiences. For example, these are 2 lectures. The next speaker, Lena Smirnova from my team, and for full disclosure, my wife is responsible for. And we have seen that in just 2.5 years, these 2 lectures have had almost 14,000 active learners when making them available online through Coursera. So there's good sides of the pandemic as well, not only the bad sides we're all suffering from. I would like to acknowledge that our work is sponsored by industry. You might take this as a conflict of interest. I see this as demonstrating that we are practically relevant in what we are doing, because it is strictly about alternatives to animal testing. It's not about their products. But we receive 3 times more money from philanthropic sources, which gives us a certain independence from industry. And our research is sponsored by the typical sources internationally, from Europe and from the US. And if you miss the logo of your company, or you have a rich uncle, I will always find space on this slide, which is already quite crowded. I also have some personal conflicts of interest I would like to let you know, so take everything with a grain of salt. I have involvements in biotechs and consulting other companies, both for microphysiological systems, pyrogen testing, artificial intelligence, and in being the editor for Frontiers in Artificial Intelligence. Okay, now let's get started. I think the important message of this conference used to be this microphysiological system we see over the last decade or more. We see that an enormous uptake in technologies which allow us to do something better. And this better is recreating the functionality and the architecture of some organs. These are 2 workshops we organized in 2016 and 2020. We published on them. Each have about 35 experts from different parts of the world stating the status of where these technologies are. And I highly recommend them as an overview where these technologies are and where they could be in the near future. The second of these workshops, published last year, just led to an article in Science a perspective article we are very proud of on 16th of September. This is a very good condensed overview about the microphysiological systems and where they are going, especially for drug development. We're going to organize a World Summit on Microphysiological Systems next year in May, June. I can be very short here because Lena Smirnova is going to give you full details on this. But this is really, I think, an important event. And one of my messages to you is I hope very much to team up more with India and other parts of Asia to make this a real World Summit. We have at the moment a very strong representation of some 45 organizations, mainly from Europe and the US. Brazil is involved, but I really would like to see also a more formal engagement and in the future, the World Summit moving around the world really as it should be. So I want to focus today only on quality. And I will talk about aspects mainly on good cell culture practice. I will touch a bit on good in vitro reporting standards, which are in their making. And I will not really talk about validation because I think most of these systems are still quite a bit away from validation, but we lay the ground by both doing our work well and reporting well on it, which is the key. So classical cell culture has a lot of problems. About 25% of cell lines are misidentified. They're often not the cells you think of. They're not the right organ, not the right species, not the right gender. And this 25% might sound shocking, but actually a tremendous number of cell lines being used are still HeLa cells. This was the first human cancer cell line in 1951 here in Hopkins. More than 20 tons of this have been grown so far. They have led to 110,000 plus publications. And you see, we are continuing to publish with this cell. About 300 per month are published. There's more than 11,000 patents with the cell line. You could easily put these flasks one on one around the world. That's the amount of cells we have grown as scientists, as a scientific community. The problem that we are often cross-contaminating our cell lines and that HeLa overgrow other stuff, especially HeLa, is very well known. These are just a few of these publications. Here is statistics I put together at some point. You see, already in 1968, the first report came out where 18 different lines, which were supposedly not HeLa cells, were all found to be just HeLa cells. And then this continues, and even in a most recent paper out of China, 2015, whereas 380 cell lines were analyzed, 95 were not what the researchers thought they are, 93% of them being HeLa cells. This is the publication. This is not to single out China. This is only one report we have in hand, which is relatively recent. And it says here very clearly, 93% of the contaminants in cell lines established in laboratories of China were HeLa. So it's quite incredible how this is persistent. Discover magazine covered this in an article, discussed this whole thing, inspired by some of our articles and discussions. So this is a cartoon which shows everything was going along fine until they discovered the HeLa cell line expressed epsilon chromosomes. It's obviously cervical cancer cell line, so female, but it's typically the opposite. You believe you have something male and you suddenly find out it's HeLa. Secondly, we are fighting with still an enormous amount of mycoplasma infection, which is not handled properly. We have all of the tools, but these are some studies. I don't want to go into any level of details, but you see here relatively high numbers in different studies over the last decades. We have to assume that still a lot of academic labs are fighting mycoplasma infection, or not fighting them, actually are facing them. And this is leading to dramatic changes in cell culture, irrelevant results coming out. Then we have genetic instability and culture artifacts in our cell lines. A cancer cell line has about 20,000 mutations on average, dramatic differences. And this was a paper already some 5 years ago we published that even in the same batch of cells, coming from one of the leading cell culture facilities, the ATCC in the US, you can have dramatic differences in the same batch of cells between different vials. And just to show you, we worked together with Brown University here. The cells already did grow differently when we took them out. The cells reacted differently to estradiol. And this also was maintained when we swapped the cell lines between labs. So it was not the performance of our experiments. They were clearly different here in the principal component analysis of metabolites formed. So they are clearly distinct cells. They show this with respect to gene expression. So there's an enormous, this is a panel of the estrogen relevant genes, and you see a clear clustering of the Hopkins and of the Brown derived cells. Still, this is a cell line published on enormously. More than 45,000 articles have been published on this breast cancer cell line and continues to be a reference point. If you do a karyogram of these cells as they come from the cell bank, you see that some chromosomes are actually hardly to identify. Others have been multiplied. There's major transpositions. So it is really difficult to call this still a cell. It is a reshuffling and reorganization of the genome. We did a comparative genome hybridization, and we found that 10% of the genome was actually lost. 50% of the genes had less than 2 copies, and 30% had more than 2 copies. Sometimes up to 30 copies. So it is really a dramatic change to physiology, and I call this a Franken cell. We have further continued with this, and we asked ourselves, how much do then at least reflect cancer cells? In this paper from earlier this year, we show that actually they don't even reflect breast cancer. They are something completely different after some 30, 40 years in culture. We compared here MCF7 cells with 1,000 different samples and with and without estradiol treatment. And we compared this with the breast invasive ductal carcinoma type of tissue they were derived from. And essentially the message is very simple. They have nothing to do with a cancer cell. So they're not a reference point for this either. And this is not singly true for the MCF7 cells. So nobody has ever shown before that the same vials from one batch of cells is affected. And most of our work in the end was done with non-passaged cells, the genome hybridization, for example, showing difference in six major chromosomes. HeLa genome has been described here in this. This is an extract from a coverage in Nature. And what you can see here, very similar, lots of different copies, rearrangements. So these cells are simply not representing physiology. This is another example which I found shocking. These are commercial cells, and supposedly they have been transfected entering a single P450 enzyme, gene from humans into this cell line. And you can see how 3 different of these products by the company are completely different to the parental cell. So you're interpreting your results as I have an additional P450 enzyme in my cell line, and otherwise it's the parent cell line. In fact, due to cloning and subcloning, you have as a consequence dramatic genetic changes between the different cell lines, and you should expect dramatic differences which are not P450 related. Another problem we too often forget is antibiotic use. This is only one of many papers, but it shows that the addition of regular concentrations of antibiotics as we use in cell culture here leads to about 5,000 different genes, differentially expressed genes in this cell line. And this is just one example. There's tons of papers which are showing how the growth behavior differentiation of cells changes with these antibiotics commonly used. So we are dealing really with something which is a highly influential factor. And there's a lot of other artifacts in the way we are culturing cells. They're typically looking like pan-fried eggs sunny side up. They have hardly any cell-cell contact. Half of the cell sees plastic and half of the cell sees culture media. This has nothing to do with the tissue organization. And we typically have a single cell type here. The density of a cell culture is 0.1% of the tissue density. So we're diluting all factors, all the communication of the cells by doing so. And now to the positive side. What has been happening by moving to the microphysiological systems is we are first of all adapted to three-dimensional cultures. And this has enormous advantages. It allows us to do many things in these cultures. It also allows us to recreate some architecture. This was an early workshop we did on 3D cultures, which again gives you a little bit of the feeling what doors are opening here with using three-dimensional organoid type of cultures. But there's also a lot more to overcome. One of the dramatic problems in our type of cell culture is the sudden changes. Because it's the cheapest and the most convenient way of culturing cells, we typically add cell culture media and every second or third day we replace it. But this means that the environment of the cells changes within a second completely. We remove all of the waste and we replenish all of the nutrients which have gone. So this is a non-homeostatic way of culturing cells. And these non-physiological environment and these sudden changes require the cells to be highly adaptable. So this is forcing them out of differentiation into non-differentiated states where they can easily react. At the same time, there's a lot of additional effects of the traditional way of culturing in a cell, from edge effects, both on the plate and in the well. There's no demand on cell function. We are overfeeding with hard to high glucose concentrations. All our cells are diabetic in essence. There's no polarization. There's no flow on these cells. A lot of things, but many of them can be overcome by perfusion. This is what an organ-on-chip system typically is offering. It also brings constant oxygen access and everything to the cells in a continuous way. And we should be very clear. Bad cells don't get better with fancy endpoints. Whether we add omics or high content imaging, a cell culture which is not good from the beginning will never result in good results. But by using primary cells and stem cell derived systems, we have the chance of really bringing good cells in. Because this was the problem so far. What human cells could you get? You could get tumor cell lines. You could get materials from the surgeon or the pathologist, but these were patients, diseased, very small amounts, and you will never get the same material again. Only with the advent of stem cell technologies, as embryonic stem cells in 1999 and as induced pluripotent stem cells in 2006, we really now have the protocols to produce essentially every type of organ and any type of tissue. And we can also then start thinking about organ interactions. One of the shortcomings compared to animals, that we can actually create bodies on chip. And the two leading projects have actually shown 10 organs for 28 days as an interactive system. So there's a lot of possibility here. We have ourselves been involved in such an organ on chip system where we helped with biomarkers, the pharmacology and kinetics, how we can model these things. But we are also interested very much in ongoing projects in how we can actually use biomarkers in these multi-organ on chip systems. And we're focusing very much on metabolomics and microRNA expression in order to find biomarkers which can help us to analyze continuously what is happening in our cultures in a non-invasive way. This is an article which is summarizing some of these problems and transitions, so I recommend this if you want a short read, moving away from culture artifacts to the new possibilities of organotypic cell cultures. So let's come to the quality assurance of these systems. And I have a long personal history in this. I started in 1996 organizing a symposium on good cell culture practice in Berlin. We're invited on this topic for the German Cell and Tissue Culture Society, and then carried it on in 1999 at the World Conference on Alternative Methods, where we called on the scientific community to produce good cell culture practice. And I had the privilege to lead until 2002 the ECWAM task force for this. When I took over ECWAM, Sandra Kucke took this, and in 2005 we published Good Cell Culture Practice. This has been cited more than 500 times, so it's a well-read article, but it is essentially saying a few things about what you should do with cell cultures. You have to know your model and characterize it. You have to assure the quality of materials. You have to document, a very key aspect. You have to think about the hazards of your work. You have to comply with laws and regulations and ethical principles, and you need proper education. And these 6 principles are laid out and recommendations are made in this guidance. But 2005 was before induced pluripotent stem cells were even reported. So we brought in 2015 an international group of organizations together, and we started with 2 workshops, one in Europe, one in the US. And we started this work. These are the 2 workshop reports where we said, how can we apply this now to stem cell derived models, to the microphysiological systems? And good cell culture practice, this is, I think, an important message. It is related to good laboratory practice, which you will know for regulatory applications, but it is not the same. Good cell culture practice wants to be applicable to all types of cell culture and especially also to academic research. So it's very basic in its way. So we created a task force, and these are some of the key people involved. Glenn Stacey, formerly at the UK Stem Cell Bank, David Pamies, at the time at my team, leading the GCCP program, and Sandra Kucke from the European Commission. And we published last year a draft which was co-authored by the steering group. This was for the last year in discussion with 350 stakeholder comments received, and we have integrated them, and the paper, the Good Cell Culture Practice 2.0 paper, is now in press and will appear in the next issue of Altex. So this is now finalized guidance which is covering or extending GCP 2.0 to the type of systems we're discussing today. So stay tuned, please. I think it is quite important that the OECD is not only aware of this, but has been adapting a lot of the good cell culture practice work. So they published good in vitro method practices, which are very much based on good cell culture practice. They're also annexing the entire good cell culture practice 1.0 and the first workshop, which was available at the time, towards good cell culture practice 2.0. So you see, there's a very intimate relation. And I hope that now GCP 2.0 will also feed into their and other people quality assurance programs. The second aspect, which I would like to stress, is it is not only about working well with cell cultures, but it's also about reporting properly about this. So in this paper, we summarize the state of the art of reporting standards for cell culture, and there's no single reporting standard available. Different to animal tests and many other areas of life science, we don't have a binding quality standard for reporting cell culture experiments, and certainly not for microphysiological systems. We also published in the same year a template for how to describe in vitro toxicology assays. Again, a big consensus process out of the EU Tox Risk project, which is demonstrating for a specific part of these things what should be considered. And this is now our next GCCP project. We have started to develop now reporting standards, and this is an open process. So if you're interested to join in, please contact me. We want to move forward here. The last point I would like to make before now coming to an end is this is all also very much related to our activities on evidence-based toxicology. Evidence-based toxicology is trying to translate aspects of evidence-based medicine, systematic review, risk of bias analysis, meta-analysis, to toxicology, because we think that it is about how we handle our results. This was based on an article we wrote in 2006 and the first conference we organized, both still when I was in ECVAM, the European Center for Validation of Alternative Methods of the Commission. And we then called on the international societies and the scientific community to create evidence-based toxicology as a discipline. And I can report on tremendous advances. In 2009, I got the first chair in the world for evidence-based toxicology at Hopkins. Since 2011, we are hosting the evidence-based toxicology collaboration. And this is an organization in the meantime worldwide promoting evidence-based toxicology. And in the board of trustees, we have the EPA represented, the FDA, the European Food Safety Authority, the US National Toxicology Program, and so on. And again, quality is key. An important aspect is to identify the quality of the data you're considering for your systematic review. I would like to refer you to this paper where we did a scoping review, showing you what is important to judge the quality of the evidence you're using for different types of toxicological studies. So here we analyzed, among others, 12 studies which were suggesting in vitro and in vivo study quality aspects. And an ongoing project of the EBTC here is what we call the in vitro critical appraisal tool, which is trying to give a tool at hand to assess from a paper, is this of high quality as it is reported and carried out. So it is a type of checklist approach in the end, which we are developing also in close collaboration with the US National Toxicology Program. So you see, quality is about quality in the lab, how you do it, quality, how you report it in your publications, but it's also key for the evidence integration, which we are aiming for in the end, taking regulatory decisions as the evidence-based toxicology collaboration is pursuing. And with this, I would like to close. As most of the time, I'm using a quote from John Maynard Keynes, an economist, as you might know. And he said very wisely, the difficulty lies not in the new ideas, but in escaping from the old ones. I hope I've shown you a few new ideas as to the quality control of cell cultures, and I hope that we jointly start escaping from the mistakes of the past.
Thank you. Thank you very much, Thomas, for this very insight presentation. Sometime I'm a toxicologist. We take refuge in the what is difference over the control. When your control is wrong, then what is, especially with cell lines, as you talked about the contamination of mycoplasma, sometimes endotoxins are also present there. They cause lots of inflammatory response. So, and we get confused with whether it is a toxic substance response or the cell's inherent response. So this I think one of the aspects which I also feel that generally we take refuge in comparing with the control. What is your control right, or is your control appropriate, and how conditions you match. So this happens, and I think you touched upon very important secretive research rather, because it's animal house, we can have this quality control and peer review and also oversight. But what we do and inside the CO2 incubator, generally we keep ourselves, and nobody has to oversee and have oversight over what kind of results are being generated, what kind of fetal bovine serum, fetal calf serum, what kind of media is supplemented to the cells, and cells with slight change in pH or in slight change in nutrients condition, it can be modulated to 1,000-fold, 100-fold, like that. So I think researchers should be very cautious about this new concept. At least I feel that this is also very reported. And also this some kind of very individualistic research. I think I consider cell culture kind of person's own, how much perfect the technicians and he himself is. So what kind of this playing with the cells, how to control that, including same thing happens with in silico, getting the desired results with the docking experiments and all that. Lots of, I think, quality control and good practices are desirable. You highlighted very important issues. So I like that you have spared some time for questions or answers. So if anybody has some query, so they may kindly put forward to Thomas. It's a unique opportunity to the young guys, young participants. If they have any query, they can put forward. I think I like this concept of good and also I think it has to be. One thing I just wanted to, so how it is possible? Sometimes the source of cells, you mentioned about ATCC. In India also, I think NCBS, we have National Center for Cell NCCS. So how much, what are the quality standards of the source of the cells? Do they follow any kind of good practices so that researchers are at least assured that whatever they are getting is the real one, and what are the simplest tools and techniques by that we can identify the cells? Can we do some genetic profiling of the cells? What are the tools available to the researchers to at least know whether the cells he is using is the right one, like that? Please, can you just comment, Dr. Thomas?
Yeah, I mean, you've now touched on tons of very important questions. Yeah, to start with the last one, I definitely don't want to give the impression that ATCC is not an excellent cell culture bank. Actually, I think they are, and you might have seen that I'm consulting them also. I just wanted to lay out that even at the highest level of quality assurance you can get, there's still a problem within one batch of cells. And the reason for this is, from my point of view, that we have actually heterogeneous populations. This is not one monoclonal cell type, but over time there are subpopulations forming. For example, we believe that there is something between 20 and 40% of MCF7 cells in our mix which lacks an entire chromosome. And this population differs in amount, and this is why also we don't get exact numbers, copy numbers. It is always a mix of these different populations, which can change probably with sedimentation of cells and depending on the exact culture conditions. There's a competition between these cells. There's a whole evolution taking place. So we have really to be concerned. There's a lot of controls introduced. Authentication of cells by the cell lines is actually pretty good. That's a very high standard nowadays. They use essentially what we use for paternity tests. So you know you have the right cell and you can check this. But it is still common practice that cell lines are passaged endlessly in the labs, that they exchange between labs. Oh, can you give me your cell line? And then the mistakes happen. And these are practices where we have to work on. And good cell culture practice is recommending them. I also liked very much when you were touching on the endotoxins, because pyrogen testing is one of my claims to fame. I've been working on this for almost 30 years now. You cannot imagine how many effects of proteins we describe in the literature are actually the endotoxin contaminations from producing them in E. coli cells or whatever. Yeah, there's so many papers around where endotoxin contamination was never excluded, and this stuff is so potent and very difficult to detect when bound to the proteins. Whenever you read this cytokine has an effect like endotoxin, you should be very careful.
Yeah. So very, very, very good topic was it for us. And if Dr. Patil, if you have questions for Dr. Thomas from other parts.
Thanks a lot, Dr. Thomas. It was really eye-opening kind of session that we had in this particular conference, because in India particularly, I'm talking about the educational institutions, we are having some shortcomings in terms of infrastructure and all. Okay, and that is where we have to be very careful. So will you give some guidelines for us academicians while taking up this kind of research, so that we can be careful when we are reporting some results from our laboratory, please?
No, I think you're completely right. I think we have to think of our papers very similar to our cell cultures. It's not about the quantity of our papers, it's about the quality of the papers. Because things which are not reproducible are simply not worth the value of the paper they're printed on. They're useless. And this is why we must assure through pressure by good peer review processes, by pressure through the funding bodies, by enabling quality assurance in the different labs, that high quality is being produced. And I think it comes with the cost. It comes with the cost of being less productive to do all of these controls. But then your publications stand. They will survive over time. They will not be shown as complete nonsense. As we have seen in so many cases, and the big discussion about the reproducibility crisis in science, I think has a lot to do with the lack of quality control, the lack of proper reporting of what we are doing.
Thank you. Thank you.
Very good. Are there any questions from the participants? If you have, otherwise, I can see Lena is there, so very much enthusiastic to speak to us. And over to Dr. Patil.
Yeah, Dr. Pratiksha Alag is already there, and she's going to chair the next session. I request Dr. Pratiksha to please unmute, and we can begin with the next session. Please, Dr.
Thank you so much, Dr. Patil. I just want to make sure I'm audible.
Yeah, you're audible, ma'am. Please.
Yeah, thank you so much.
We
Very
can
good evening to one and all. Yes, good evening, Dr. Raisuddin. How are you? Yeah, so thank you so much, Dr. Patil, for having me chair the next session, which is the 6th session for the 4th annual conference of SAAE-INDIA. A very good evening to everyone present and attending this conference from India. And a very good morning to Dr. Lena Smirnova from CAT John Hopkins. I know it's early morning there. So thank you so much for accepting our invitation to speak for session 6. I would also like to extend my thanks to Dr. C.R. Patil and the respected members of DPSRU to having given us the opportunity to conduct the session, and also to Dr. Akbar Shah on, for having us on this conference. So to begin with, I would like to introduce Dr. Lena Smirnova. She is from 1997 till 2002, she was completing her master equivalent diploma in International Sakharova Environmental University at Belarus, followed by her PhD with a focus on neurosciences from Charité University, Berlin, Germany. She is currently a researcher at the Center for Alternatives to Animal Testing, Johns Hopkins University, and she's heading the education program and the program on microphysiological systems, with her research focus on gene-environmental interactions in autism and using stem cell-derived brain organoids. She has completed her postdoc at the Center for Alternatives to Animal Methods with her project title on role of microRNAs in developmental neurotoxicity. I welcome Dr. Lena to today's conference. Her title for today's conference is iPSC-derived brain organoids for developmental neurotoxicity testing in the 21st century. Thank you so much. I'll now request Dr. Lena to please take over.
Thank you very much. Can you confirm you can hear me? Yeah, perfect. Okay, so thank you again for this kind invitation. I am humbled to talk today and follow my presentation of my husband. So thanks for a very nice introduction. I will jump to the next slides. We will be talking about developmental neurotoxicity of this century. The first conflict of interest disclosure, the model I will be describing today is licensed to the bio company called Axozim, and I'm consulting Axozim in their research. So let's start from the beginning, and let's start talking about chemical testing and where we are in general with chemical testing. Worldwide, we have over 100,000 chemicals in consumer products, and only in US we have produced 1,000 new chemicals every year. If we would be now in person, I would ask the audience, what do you think? What is your guess? How many chemicals we have tested for their toxicity properties? Since I'm online, I cannot ask that. I will tell you that it's very little. We have only 7% chemicals extensively tested, and we have 60% of the chemicals we have some type of data on them. And this is only after the REACH legislation reached the deadline in 2018, which is the European legislation for registration of the chemicals. So you can see that from these hundreds of thousands of chemicals, we know very little about toxicity of those chemicals. And when we talk about neurotoxicity, it's the problem is even worse. So about 1,000 chemicals from the chemical universe were tested and shown neurotoxical potency in experimental setups, and only 200 were shown to be neurotoxic in humans. And when we talk about developmental neurotoxicity, we have only a handful number of chemicals, precisely 14 for now, which are confirmed developmental neurotoxicants. And it is not because the rest of the chemicals are safe. It's just because the rest of the chemicals were not tested for DNT. And now I will explain you in the next couple of slides why. So in general, when we talk about the toxicity testing, a lot of methods which we use are really out of date. So if you look in this slide, our LD50 test was developed in the 20s, chronic toxicity in 30s, iron skin irritation in 40s, and reproductive toxicity in 60s, following cancer bioassay in 70s. So those all assays were developed before I was even born. And when we talk about developmental neurotoxicity, keeping in mind in the background that over 90% of clinical trials are failing, and if we talk about the brain, for example, Alzheimer clinical trial, the ratio is 95%, and we still don't have a cure for Alzheimer. And also productivity between different species is the problem, specifically for such complex endpoints as developmental and reproductive toxicity. Mouse would predict rat only by 65%. So, and then we have a problem with DNT testing that first of all, the animal is gold standard. For those, we were following OECD test guidelines to do the developmental neurotox testings. There are ethical concerns for that. They're very expensive, time and labor consuming. And therefore, DNT and DNT tests and also neurotoxicity tests are not mandatory. So they're only requested if we see a certain red flag in other tests which are conducted before that. There's one exception. US EPA requires to test for neurotoxicity all pesticides which are on the market. So from that reason, we have not many chemicals tested for developmental neurotoxicity. And also, as I just said, mouse doesn't predict rat very well. So why we should assume that mouse or rat will predict human? So we have different comorbidities. We live different lifestyles as the inbreeded mice in the cage. Ethnicity, gender, and so on, which plays a role. So that's why we have all those issues with the current approaches. And in 2007, or prior to that, but it was published in 2007, the researchers came together. It was actually requested by EPA, US EPA, to rethink the toxicology paradigm and then revolutionize toxicology, to move away from black box, time-consuming animal experiments and use the tools which we have on hands, and because the life science is developing really fast, and use those tools and apply them for toxicology, use modern technology, human relevant model, and high throughput testing. And this will allow us to test more chemicals faster and cheaper. And this initiative was supported by many agencies, especially in US, but also in Europe. You can see here all the main key players in regulatory toxicology are here. NTP, National Toxicology Program, EPA, FDA, NIH, and DARPA. And really interesting projects were developed by, from this initiative of NRC report. You might have heard about ToxCast and Tox21, which is the high throughput screening programs running in US, NIH, and EPA, screening thousands of chemicals and hundreds of the assays, predicting the toxicity, and which accelerated testing a lot. And this is also support on the regulatory level. You might have heard about legislation in Europe, which is REACH, Regulation, Evaluation, Authorization of Chemicals, which is active since 2007, with the deadline 2018, where all chemicals which are produced in Europe over 1 ton should be submitted at a year to the ECHA chemical agency in Europe. So the regulated industry is forced to to accelerate the testing, and they automatically should use alternatives. The same in US, the reauthorization of Toxic Substance Control Act, which was signed by Barack Obama in 2016, together with REACH, they enforce risk-based chemical assessment, monetary evaluation of existing chemicals, more public transparency, and they automatically promote new approach methodologies. So what are those new approach methodologies? Yeah, there's many of them, and we can talk about computational toxicology, meta-toxicology. Today, we'll focus on microphysiological systems, or MPS, as a part of new approach methodologies. And our vision is, yes, we have to replace animal testing, but we cannot replace them with a simple two-dimensional cell cultures. And partially, you might heard from Thomas an hour ago why the cell cultures have, which problems we are having with cell cultures, artificial cell lines and monolayer cultures. So therefore, our vision and our goal is to replace the animal testing with microphysiological systems, so advanced cell cultures, let's say. But it's also not enough. We need to apply also computational bioinformatical tools and apply systems biology and toxicology approaches in combination with microphysiological system to get a full picture what is going on mechanistical level to apply to perform mechanistical toxicology. So what is microphysiological systems? Microphysiological systems is an umbrella term, and what is recapitulate organ or organ functional unit. And the most important, microphysiological system recapitulate the functionality of the organ. And there are different types of it. Yeah, so it's microfluidic system where you have a perfusion and stimulating the blood flow, for example, or movement of the organ, for example, a lung or heart in organ on a chip system. Body on a chip, it's a combination of different chips together to recapitulate many organs together. For example, you will have liver to metabolize the chemicals, kidney to excrete the chemicals, and then heart to pump the blood around, and so on. And those studies are ongoing in many different laboratories. 3D cultures, organotypic cultures, again, recapitulating the architecture and functionality of the organs, spheroids and organoids. And Thomas mentioned it already. So we met in 2019, it was in a workshop in Berlin where we discussed where we are with microphysiological systems, and it was nicely covered recently in Science. So if you want to have a crisp summary of this workshop report, please follow the Science article and you can see where we are, what are the challenges, and what are the next steps in microphysiological systems. And again, as Thomas already mentioned, we are organizing. Actually, at that meeting in Berlin, we discussed with main stakeholders that we need to have an annual meeting to discuss these fast developing tools, and also we need to have a society around that. So, and this is therefore we established we are establishing, it's not established yet, a series of MPS World Summits toward formation of the MPS International Society. And we had so far 2 virtual events, and the first meeting will be in New Orleans in 2022. We have a steering group formed, we have a scientific advisory committee where we have funding, secured funding from NIH and CATS through the grant. We also, as you can see on this slide, we already have some sponsors committed toward the first summit, gold, silver, bronze, and exhibitors. And again, if you're interested in support this initiative, so your logo can be here. Briefly, what we discussed in our virtual pre-events. So the first virtual pre-event was focused on MPS toward regulatory acceptance. So what should be done to harmonize, to validate those MPS technologies to make regulator accept them. And you can see the panelists. We have presentations from young scientists, and we have a panel discussion. You can see the lineup of the panelists, really key players from different regulatory agencies. And a second event, which just took place this Thursday, we focused on the new developments of the cell culture and also bioengineering part of it. And again, it was really nice attended. We had over 570 registrants and over 300 people were live attending that, live on Zoom. And again, great lineup of the panelists discussing the MPS. So here, mark your calendars for June 2022. I hope you can make it to New Orleans, a great city with jazz music, to learn even more about MPS in person. And here I just listed some themes which will be covered in this 4.5 days conference. And below you can see the link where you can find more information. So now let's move forward to brain, and talk about brain microphysiological system, specifically about brain organoids and how we can use brain organoids for developmental neurotoxicity testing. So this is the brain organoid cartoon, which was recently published, actually also by my co-workers Thomas Hartung and Helena Hogberg. So you can see that this is a very versatile tool. We can use brain organoids to study different cell types of central nervous system, neurons, astrocytes, all the supporting cells. We can study histoarchitecture, synaptogenesis, functionality, and communication. And also we can look into the drug treatment and screening, as well as toxicity testing of the chemicals. By putting the brain organoids on the multi-electrode array, where we can measure the electrical activity in the brain organoids, we can study the functionality of the systems. Historically, our lab started to do three-dimensional cultures when Helena Hogberg and Thomas Hartung established the lab at CAT, which was around 2009. And with the secure of NTP and FDA support, financial support, Helena adapted RAD 3D reaggregating cultures in our lab to study flame retardants. And she used omics technologies to analyze the network and perturbation signatures after the exposure to flame retardants. So why flame retardants? At least in the US, flame retardants are added to every piece of furniture and mattress, and even to pajamas of the babies, to reduce flammability of these pieces of furniture or clothes. But those chemicals are not, they have so much, their amount is so high in our furniture, but they're not properly studied. And recently, the original polybrominate flame retardants were replaced because of the concern to be developmental toxic. They were replaced by organophosphorus flame retardants, which don't have much data either. So we have a lack of hazard risk assessment on these chemicals, and we're still putting that in our furniture. And to be noted here, those flame retardants, organophosphorus flame retardants, have structural similarity to organophosphate pesticides, which are by design neurotoxicants. So Helena and her team looked into the transcriptomic and metabolomic signatures in this rat mini brain model after exposure to the organophosphate flame retardants, and they found the alteration in gene involved in neural morphology, reduction of neurotransmission, synaptogenesis, and alteration in glia-specific genes. They also found a neuroinflammation response. And if you need more information, please follow the link. It was recently published in Archives in Toxicology. Well, this was a rat model, and this is a great model, but it's derived from rat pups, So we decided to humanize this model by using induced pluripotent stem cells, so IPS. And this was done, this grant was received by Thomas and Helena from NIH NCATS, and David Pamirs, who's here in the picture, was conducting the experiments where we differentiated IPS into the neuroprogenitor cells first, and then we put those neuroprogenitors in the single cell suspension on a shaker. And due to gyratory shaking, they form these tiny, tiny little dots, as you can see in this picture, in a 6-hole plate, and those are brain organoids, which you call brain spheres. So what are the brain spheres? They have also types of the central nervous system, different types of neurons, astrocytes, oligodendrocytes. They don't have microglia, but we can add it, and we have experiments done on that already. What makes our model particularly interesting is that it has myelination. Around 40% of axons in our model are myelinated. It's very controlled in terms of the size and the cellular composition. And since we have IPS as an original cell, we can use different genetic background for those brain spheres. And this is the short summary of all different kinds of projects we are working on together with our collaborators within Hopkins, outside Hopkins, using this model. I'm not going to talk about all of them. I will focus only on DNT as a topic of my talk and briefly touch on gene-environmental interaction. So again, how can we replace animal testing for DNT with in vitro? It's a difficult task because the brain is the most complex organ in our body, right? So, and then the experts in the field of DNT came up with the battery of tests, or vitro test battery, which will cover key events of neural development, right? So we have a cellular events such as proliferation, neuronal differentiation, glial differentiation, migration, synaptogenesis, electrical activity, neurite outgrowth, and so on. So if we can have a battery of tests which will tackle all those key events together, we can assess, at least prioritize the chemicals and assess the chemical potency to be developmental neurotoxicant. So, and this is a table from the publication, which I referenced to the left, showing that there are different tests already developed. And this look for you as numbers, or there's random numbers and letters. So this column shows name of the test, and it means it was developed in different cell types, in different laboratories. Some of them are rodent, some of them are human. And this letter code and color code shows you the readiness of this test, how ready it is to be implemented in DNT battery of tests. And you can see that some of them are still not ready, including human three-dimensional cultures, myelination, synaptogenesis, and so on. So looking at that table and looking at all this diversity of the tests which are already there, and looking back to our model, we thought, wait a second, we can cover with our brain organoid model most all of those key events which are needed to prioritize the chemicals for DNT. We have neurodifferentiation, we can study myelination, neurite outgrowth, synaptogenesis, glial migration and gliosis, network formation. Then the next question was, how can we do that? How can we do that? We can do, of course, we can do the imaging, we can do the staining, we can do PCRs, but how can we do that in a high throughput manner? How can we test a lot of chemicals using that model? And as already was mentioned in the previous session, timely with our thoughts about DNT battery, US EPA announced that they are going to reduce use of animals for EPA needs by 2025 and eliminate animal use by 2035. This is a really ambitious goal. And in line with this goal, they announced also grant support to advance research and alternative to animal testing. And we were a lucky group to receive one of the 4 grants, and I'm leading this effort. And you can see all my co-workers and co-PIs from our center, but also we are teamed up with School of Engineering and Bioinformatics and also School of Medicine. And with this project, we are developing a multiplex human breast sphere developmental neurotoxicity test for 6 key events of neural development. So how we want to do that? We want to create a mini brain ball, if you wish. So we are using CRISPR-Cas9 technology to fluorescently tag different lineages in our brain organoids. So we develop an IPS line, which will have, for example, fluorescence tag behind tubulin promoter, green fluorescence tag behind PLP promoter, which is oligodendrocytes, red fluorescence tag behind astrocyte, GFP promoter, and so on. And then we differentiate these IPS into brain organoids, and they will lighten up with different colors so that we can follow differentiation live and stages of differentiation, how synaptogenesis occurs, how oligodendrogenesis occurs, but also we can screen chemicals using high content imaging. And here are some examples which we are already succeed doing that. So this project is in progress, and as I just said, it's supported by EPA and also SOT Colgate. So this is the first fusion cell line we generated, which is GFP PLP, and PLP is a marker for oligodendrocytes. So you can see nicely here the green endogenously expressing GFP oligodendrocyte, and in purple you see the axons. And also here in the flow cytometry plot, you can see that our GFP signal is nicely colocalized with the antibody staining against PLP. This is another example where we colocalize endogenous PLP, green colors with MBP marker for myelin. The next construct we designed is synaptophysin. In this case, it's also green, but we are also working on a different color to incorporate in PLP cell line. And you can see here in this video, immature 2-week brain organoids, which are expressing green and synaptophysin, and red MAP2, which is the dendritic marker. And this is more mature cultures of 6-week organoids where we have much higher expression of GFAP. Sorry, it's not GFF, GFAP. So showing that we can follow synaptic formation over time. Also, as a part of this EPA-funded research project, we are developing a new 21st century electrophysiology. So originally, electrophysiology is the plates with the electrodes where you can sit your cells on top of the electrodes and record the electrical activity. But it doesn't work very well for 3D because the 3D will the density of the electrodes is very low. It will touch brain organoids only in one single spot. So therefore, together with our collaborators in School of Engineering, David Gracias and Ki Huan, we're developing EEG for our brain organoids, if you wish. So such cages which cover, as you can see on this cartoon, they would cover the brain organoids from each size, and they are populated with the electrodes where we can, as you can see here, we're already recording the electrical activity from those. And this is the design of the cages, and then this is how the brain organoid is sitting in this cage. So now I will talk a little bit about different projects on DNT, how we use the brain organoids for that. So the one was we studied the nanotoxicity, not necessarily DNT, but also in general neurotoxicity, how different nanoparticles with different coating may affect the our brain sphere model. And I'm not going into details. You can see here in green the pegylated nanoparticle. I'm sorry, within the brain organoid, and this work was published mainly in Fibers, and then was mainly done by Paolo and Mariana. We also looked into the developmental neurotoxicity of pesticide Rotanon, and David and Katarina were mainly working in that project. And what we found is that the younger brain organoids are more sensitive to Rotanon than the mature cultures. And we can also look, the Rotanon is a specific, specifically killing dopaminergic neurons, which we can also observe in our cultures. We looked into DNT effects of antidepressant paroxetine. Paroxetine or Paxil is an antidepressant used for postpartum depression, also by lactating women, but also has been extensively used during pregnancy. Also used a number of disorders related to anxiety, menstruation, and menopause. And what we found is that paroxetine reduced the neurite outgrowth. So this is the assay we developed with the brain spheres. So basically we plate the brain spheres on the plate, let them attach, and when they have the matrix they attach to, they start to let the neurite to grow out to look for neighbor cells, to look for new connections. And then you can measure and quantify the length and density of those neurons with high content imaging. And then you can compare how the chemical or drug treatment can affect that. And you can see that the physiological relevant concentrations which were found in the blood of women from the literature was already reducing the neurite outgrowth in our system. And also it seems that it's increasing slightly the GFAP, suggesting that we have probably gliosis going on. We also looked into synaptogenesis after long-term exposure to paroxetine, and you can see here we quantify the synaptic pointer in the cell cultures, and if you compare that to the control, you can see significant reduction by paroxetine, 20 and 60 microgram per milliliter, and which was comparable with the clopidogrel, which is non-chemical to affect synaptogenesis. As already mentioned, our model has myelination. And then we said we need to use this opportunity to establish a myelination assay. You can see here the electron microscopy picture where we have a nice wrapping of myelin sheets around the axons. And what was done mainly by Megan and supported by Serjan and Helena and money support from Roche and Colgate, we established the assay where we can quantify myelination. So you can see here myelin-based protein staining in control treated samples and samples treated with cuprizone, which is a non-demyelinating agent. And you can see how the myelination is going down while the axonal staining is still present. So, and by overlapping these two, we can quantify the myelination percentage in different cultures upon the chemical exposure. And this work was also recently published by Megan. So follow the link to follow this publication. So in the last, I would like to talk briefly on how we can use brain organoids for neurological disorders and specifically for autism. I'm interested very much in autism and especially in gene environmental interaction for autism. Why? So yeah, you might know that autism prevalence is strongly increased over the last decade. And now US Center for Disease and Prevention, Control and Prevention, CDC, report that 1 in 54 children born in the US is diagnosed with autism. So this number and this huge increase can be explained only by better diagnostics. It can be explained by difference in genetics in such a short period of time. Of course, there is a huge component of genetic component and heritability in autism, but environmental plays also a role. But there is no single environmental chemical which was absolutely confirmed to cause autism on its own. So therefore, our hypothesis is that it's gene-environmental interaction. So you have a certain genetic predisposition to develop autism, but only in combination with certain environmental stress, you will develop the disease. It will trigger the disease. Or if your genetic predisposition is strong enough to have a disease, with the different exposures or environmental stressors, you can aggravate the symptomatic and put the people in different spectrum of this disorder due to environment. So, and then we wanted to study the gene-environmental interaction in autism, and then we discuss where can we start? The autism is such a multigenic disorder, so it's hundreds of genes which are associated with autism. Where should we start? And we decided to look into the high-risk autism genes. In particular, we decided to look into the CHD8 protein, which is the chromatin remodeler, and it's expressed early in development in the midterm of the pregnancy, which is nicely covers in our brain sphere model the stage of development, and it's regulate the chromatin structure and therefore has a lot of targets. And this is then here on the slide you can see the network of CHD8 target genes. And interestingly, a lot of those targets are also autism risk gene. He highlighted his in black. So being itself an autism risk gene, it regulates also a lot of autism risk genes. And as an environmental exposure, we decided to look into chlorpyrifos. It's a very debated compound. It was finally banned in US by US EPA only this summer, or after years of discussion. Chlorpyrifos is suggested to be developmental neurotoxic and lead to the neurological perturbations. So this is two components we have: CHD8 as a genetic background, and chlorpyrifos and its active metabolite oxone as the chemical exposure. And to do so, we team up with Dr. Herbert Lachman, who developed the IPS line where he knocked out the CHD8 gene. So he developed a heterozygote knockout, CHD8 plus minus, and we received this cell alongside with the control cell line CHD8 plus plus. Sorry, it's a mistake on the slide again. So it should be knockout as a second line. So, and we generated the brain spheres from these control and knockout cell lines, CHD8 plus minus, CHD8 plus plus, and we exposed those brain spheres to chlorpyrifos and chlorpyrifos oxone. And then we looked in different signatures, molecular signatures and functional assays to see whether we have a synergy between whether the mutant cell line is more sensitive to chlorpyrifos than control cell line, and where the synergy between two of them. And this work was done by Kshilitsong and Sergio Mantaferri, and was recently published in EHP and funded by Alternative Research and Development Foundation. So the first thing what we looked at, what is the level of CHD8 protein in the cultures? And then as expected, we saw, so this is the control cell line. And if you compare the level of CHD8 in the mutant cell line, obviously it was lower because it is a knockout. But interestingly, we also saw the huge reduction of the level of CHD8 also due to exposure to chlorpyrifos in both control line and in the mutant cell line. So this may suggest that the chlorpyrifos can have the same downstream targets and affect the same pathways as CHD8 mutation. And this is one of the main synergies we found. And another big work we've done, we compared the signature, molecular signature, metabolic signature in our system with human existing data. So we looked for different neurotransmitters which are known to be perturbed in autism, different metabolites tackling different pathways, energy pathway, carbon metabolism, and so on, tryptophan synthesis, and so on. So, and we looked into the level of those neurotransmitters metabolites in the blood from autism patients. We didn't look that, we looked from the literature, in urine and in the brain. And then we compared the levels of those metabolites and neurotransmitters in our brain sphere system, and what was the changes due to mutation, due to the exposure, and whether we found any synergy. So in this table summarized that, and you can see that for some of them, we did find a synergy. For example, if you look into tryptophan or its metabolite cunuric acid, we had a slight increase of cunuric acid already due to mutation, which was much higher when we exposed the mutant cell line to chlorpyrifos, suggesting a synergy. Or again, with tryptophan also, we saw the effect on tryptophan only after exposure of mutant cell line to chlorpyrifos. Huge synergy in L-alpha hydroxyglutaric acid and also in lactic acid. So suggesting that there is a certain aggregation of what CHD8 mutation does to the neurodevelopment. We aggregate that with exposure to chlorpyrifos. And with that, I would like to conclude and say a huge thank you for your for listening to my presentation, and I'm happy to answer any questions. And here I posted a picture with a current group who is working currently in our lab, and this is our current DNT group, which of course it's changing. So a lot of people are behind this work. Thank you very much for your attention.
Thank you so much, Dr. Lena, for an amazing lecture. I think this was definitely an eye-opener for all of us to know the total number of chemicals that are truly tested end-to-end for toxicity. I mean, those figures were really astonishing, with only 16% actually going for end-to-end toxicity. I'm sure this has made me aware of the total unexplored area and an ignored area, which is important in toxicity, that is the neuronal toxicity. And because it has a very delayed impact, on the growth and development of human. So I think that is something we should really consider, in great depth in the future. Most important thing that we've really highlighted is the lack of any representative human model already available for studying these kind of developmental neurotoxicities, which is, again, a great area where all of us as scientists should really put in our efforts to and develop such human model. The most important thing is the complexity of the neural network and the cost that goes into these kinds of experimental designs, and the technical difficulties that we face is something that was really very well highlighted in your talks, and definitely the impact and the significance of the MPS. So thank you so much, but there are certain questions or there are certain clarities that I would really like to address with the kind of talk that you have told to us. So first thing that I would really like to ask you, Dr. Lena, is unlike any other organ of the body, the brain is very well protected with the blood-brain barrier. When you talk about these development of these organoids, where we don't have that kind of a blood-brain barrier, which is restricting, which is a natural barrier by nature, which is restricting the entry of these chemicals into the human brain. How well do you think that these organoids can be a representative of the actual human toxicity that is coming out from the exposure to these neurochemicals? So, I mean, what kind of a correlation would you like to draw with the actual brain, which is protected by the blood-brain barrier or by the presence of the CSF, to these organoid models?
Yeah, this, of course, is a very important component of that. And there's a lot of work. We also had some experiments toward generating the blood-brain barrier, and a lot of work in different groups developing the barrier on the chip and blood-brain barrier in particular to address exactly those questions. And also for pharmaceutical industry, it's very, very important and interesting aspect, because they want to have from different angle, right? It's not from a toxicity angle. They want their drugs to enter the brain. A lot of chemicals are actually passing the blood-brain barrier. For example, like chlorpyrifos or lead, and also another point, we are looking into developmental neurotoxicity, which has different stages. So we always should keep in mind which answer my model can give me. And we are looking into embryo and fetal development, where the blood-brain barrier is not formed yet properly. So we are looking into where this is not functioning as supposed to function in an adult organism.
Okay, so I think that is very important point that you have made, that you're looking at the developmental neurotoxicity, where we have not seen, where the fetus is under the developmental stage. So on that front, one thing that I would really like to ask is the study that you have shown where you have used chlorpyrifos and at different concentrations. Is these the concentrations, the representative concentrations of exposure, or the concentrations that are really, where we are really exposed to? Because when you talk about these kinds of, say, pesticides or any toxic chemicals which are in the environment, their concentration is actually the one that is the most detrimental factor that decides the neurotoxicity or any kind of toxicity for that matter. So how have you really worked out on finding out the concentrations of these chemicals, and your experimental design? Has it been focused more to the realistic concentrations of exposures, or are they just because you want to study the effect of the toxicity that you've used a high dose concentrations to really understand the impact, the genetic impact?
Right. Well, in our lab, we are doing actually both approaches. So in this particular project on autism, we used very short exposure, which was only 24 hours. Therefore, we had to do high dose, which is not a relevant dose, that's for sure. But this was just a model to see which toxicity pathways we can induce. But we have a lot of work also on, we are looking into the more human relevant exposures. For example, we have now work ongoing with support from FDA on metal toxicity, and in particular metal mixture toxicity, where we're working with the concentrations which are on water level, accepted water level by EPA. So this is the concentrations we are using in our system, and also which are actually lower than, for example, Superfund sites. This is certain areas in US defined as Superfund where you have accumulation of certain chemical. So for example, we know that we're working with concentrations which are below blood level, for example, talking about lead, and that's a particular aspect. So for example, we're working with lead concentrations which is below concentration which is found in blood in children from Superfund sites. So we have, for example, paroxetine I've shown. This was also physiological relevant concentration. We performed a literature search and found what was found in the blood from women who was taking this drug, and we applied this concentration. But this was long-term exposure, so we exposed them for an entire period of differentiation for 8 weeks. So that's why with chlorpyrifos, autism project, since it was short-term, was not physiological relevant concentration. So we do both, and we very much aware of the limitations using the high concentrations. And for example, if you go to the publication, you will see that we are not criticizing ourselves, but we are pointing that out. Yeah, so that you should be taking that understanding that it's not what we find in the environment.
Yeah. Okay. So yeah, I think thank you so much for clarifying all my doubts related to the study and the use of organoids, brain-derived organoids. I'm really impressed by the kind of work you have put into, supporting the cause of alternatives and trying to promote a more humanized or human-centric approach towards a disease and the use of non-animal methods. I don't have any other questions, Dr. Patil. If there are any other questions that need to be answered by the other members, I'll be happy to take them.
Yeah, Dr. Leena, there is one question from audience who are attending through YouTube channel. There is Mr. Shivam Pandey who wants to know whether we can develop midbrain organoids. He wants to know whether there should be region-specific organoids, and then can we develop such organoids for Parkinson's disease? And is it possible to detect the neurotoxic agents which cause neurotoxicity to midbrain? And then can we track the progress of Parkinson's disease in that particular model which we will be creating? That is the question he has asked.
Yeah, so to answer the first question, yes, you can develop a midbrain model, and there are other such models, and there are groups which are working, for example, Dr. Song, who is very much in IPS field. So he has, for example, different regions. He developed different regions, including the midbrain. Dr. Praska also group has different regions, and also there is a next step of the brain organoids, which are called assembloids, where the people develop in different regions separately. So you will have a cortex organoid, for example, and another region organoid, and then they put them together. And, or, for example, you have organoids with predominantly excitatory neurons and then predominantly inhibitory neurons. They put them together and let them migrate to each other, and it's called assembloid. So this is like a next level of complexity, a next step to that. In terms of the Parkinson's disease, one might wondering, oh, actually, so the neurodegeneration, you're having developing model, right? So our model represents embryonic development. We are not on the level of mature brain, obviously. But people who are doing, and there are a lot of groups working on neurodegeneration, Alzheimer's and Parkinson's. And people who are working on that, they argue that development and neurodegeneration has very much in common. So you can, they apply also, they're trying to senescence the model, but also still using that model, you can answer a lot of questions for neurodegeneration. And for example, as I showed you with Rotanon, we had also different studies where you can, using the brain organoid, specifically kill only dopaminergic neurons and then study Parkinson.
Thanks a lot, Dr. Leena. Dr. Pratiksha, we can conclude this session with your remarks now, please.
Thank you, Dr. Patil. So first of all, Dr. Lena, thank you so much for this session. It was a beautiful and an interactive session, a great insight into the development of brain organoids using induced pluripotent stem cells. I thank the organizers of SAAE-INDIA for organizing this beautiful session and giving an opportunity to all of us to be present over there. I thank the members of the DPSRU University who have taken the charge of the smooth conduction of this conference. Thank you so much, everyone. It was nice to be a part of this conference. Dr. Patil, I'll hand over to you. Thank you.
Yeah, thanks a lot, ma'am. I think Dr. Akbar Shah sir is there. I request him to unmute and please take charge of the dais. Dr. Akbar Shah sir, are you there? Hello. Okay.
Coming, Patil, I will just join. Yeah. So,
yeah, thank you.
My dear speakers, chairpersons, and participants, I should say good evening to all from India, and even to those coming from Europe, and good morning to those who come from USA. And we have had a very wonderful day and a session. Though we started in the afternoon at 2 o'clock after lunch, we went up to this time. It is now it is almost 7:30, and 8:30, right? Yeah, it's 8:30. So the whole day, 6.5 hours, have been very rewarding exposure to various facets and issues in alternatives to animal experiments. We have had a very important and interesting inaugural session with Dr. Raisuddin initiating the program with his brief address about the way the society was started and how it has been functioning, and what is the stage at which society is now and what it is expected to take to. Then as a very senior person, very conversant and communicative. Yes, he could do very well introducing about the society. Well, I of course did give a brief introduction about the background of the society and the way and why it was started and how it was going, and also about a brief introduction about the proceedings expected of the day. And Dr. Dr. Goel, the Honorable Vice Chancellor of Delhi Pharmaceutical Sciences and Research University, very simple, very simple, I would say. He could very easily communicate with the people. He has a lot of things to speak about alternatives, both from the positive side and a little bit on the negative side also. As a faculty and a scientist of pharmacology, I am aware for several years that the pharmacology people have some issues with regard to banning animal experimentation, at least at the postgraduate level. Well, he didn't hesitate to say that, and I think the regulatory authorities, the Pharmacy Council and the Medical Council are aware of that, and until now, the PG studies, the experiments are not dropped yet. Well, we had Dr. Gupta, our president, giving a very elaborate account about the state of affairs with regard to alternatives, and he was also talking about the pros and cons, and very emphatically he was saying that society is not opposed to animal experimentation, and it is subscribing to the 3R concept. So where there is no alternative, replacement alternative, we go for refinement and reduction. And when we don't have any of these three, then we'll have to go for animal experimentation until adequate or suitable alternative becomes available. So it is not anyway a war between pro-alternatives group and on the alternative group. We go a tightrope walking, propagating science of alternatives, and there's no conflict. Yeah, he was very clear about it. And then, of course, we had the scientific session starting. We had 6 wonderful speakers, and of course, the choice was mine. Basically, I should own the responsibility for choosing these as the speakers. And of course, it played a little role on the choice of the chairpersons as well. I gave some freedom to Dr. Patil to choose who would chair which session. So we had 6 wonderful speakers, and the science they were communicating was really very brilliant. I need not be highlighting that each one was very special in his or her own domain or talk. I'm not going to dwell into the content of each one's talk, but I am more concerned about the objective of the society and how, to what extent these deliberations, the 6 speakers and the chairpersons have contributed to the objective of the society. Well, the society has been, out of my experience, when some of the senior colleagues, exposure to the alternatives across the globe, we were much particularly concerned about the very slow progress these alternatives was making in India. So that was our concern. So we started the society, and the idea was to sensitize our scientific community, the students, teachers, scientists, and the industry, to the various alternatives available and what kind of progress is made in the developing countries compared to what progress we are making. So that has been the major objective. I'm very happy. My colleagues, the senior colleagues, have been supporting me, and many have joined in this venture. And these six speakers they did not disappoint in efforts. They were more than what we were expected of them. Starting from Dr. Pant, a senior scientist from IITR, he has been giving you overall coverage of what has been happening in the IITR with regard to discovery of newer alternatives and implementation of alternatives. And that was an eye-opener for the younger scientists, younger students, the scholars, what kind of facilities available in IITR, what know-how is available. Well, they can definitely avail the opportunity of interacting and availing the facility at IITR. We had a young speaker, Francois Pascate, coming from Belgium. He is also known to me for a long time. Very, very enterprising person. He was like us, basically a scientist attached to one of the university system, and later, after realizing that there is a lot of more scope available for expanding the horizon of alternative service, particularly the European Union, he took to a kind of a voluntary organization kind of Ultratox, and though he is still attached with the CART EU, is Center for Alternative Animal Testing Bureau. He is also doing freelance assignment, training, and taking up projects and cracking issues. Very, very nice person, and he could speak authoritatively when things happening in the Europe. We had Dr. Adrian Smith, who is again known to me for several years, ever since I got into this domain. And particularly, he got me attraction when he was recently World Congress of Alternatives. He was conferred an award. So he caught my attention, and therefore I invited him, and again he was available, and it is very nice of him to have participated in this. Well, I am always attracted by his approach to animal and non-animal methods. He used to say, arrive, inspire before arrive, prepare before arrive. Okay, so these are very, very, very excellent slogans or expressions that make you prepare for good experimentation. And he threw open ideas that we have opportunities to interact with the Noricopa, and so it is open for young people and even the society. I have already alerted one of our senior colleagues to associate and interact with the Noricopa so that the society is linked with them. And Mohanan is again a very senior scientist coming from Sri Chitratarna Institute for Research, and again known to me for a long time. I very deliberately invited him because I knew very well that he has a huge grant coming from the Department of Science and Technology, very specially to develop a human on chip or organs on chip. And knowing very well his contribution to science for alternatives, particularly organ on chip, I invited him, and he did not disappoint us. He did very well to apprise the listeners, participants as to what is actually organ on chip and what is the modalities that are adapted, practiced to develop the organ on chip, and how, in what way the organ on chip is going to be useful and applicable. Brilliant talk. And then we had, of course, the two very, very special people to me, Thomas and Lena. They are husband and wife. I knew very well, and that's the way I invited them. So Thomas is in his own way. I am an admirer of Thomas. I have nothing to conceal. I am always overwhelmed by the way he handles the subject, and particularly what he spoke today is close to my heart. A good cell culture practice, because I use cell culture extensively in my research until today and in few years to come. And of course, I am aware of the problems he was highlighting, but the way he was explaining it, well, it's an eye-opener for all of us to be consistent with the kind of quality of the cell that we use and the quality of the experiment that we conducted. Real good science. And Elena, I know her as a postdoc when I had been to the CART, Johns Hopkins, and when they were, she was and her team was doing work on this brain organoid. I have seen them really doing this investigation, and now she is transformed and metamorphosed into a scientist of very high caliber, from a postdoc to a scientist of caliber, and a wonderful science has come out from her. From what we listened to Mohanan about the organon chip, the introduction to the concept, we have heard from Lena how the organoid chip is really in real world situation, how it was developed, the brain organoid, and how the brain organoid could be of application in neurotoxicity testing, and the different trends in terms of the brain organoid development and the newer versions and applications that are coming up. So I cannot take any more time to stand between you and your dinner. I thank each one of the speakers on behalf of the organizing committee and of course the society and the particularly on behalf of the Vice Chancellor, Honorable Vice Chancellor of Delhi Pharmaceutical Sciences Research University. And I thank the chairpersons very profusely, but keeping up the time and for the nice kind of introduction they gave about the speakers, and the good discussion I appreciate. Particularly the last two speakers, there was a lot of points raised and they were able to answer the questions. And that should be the way tomorrow, perhaps. There should be a lot of points raised so that issues that are still remaining unresolved will be clarified. So that is what they lead and guidance for the future. You may post the questions in the chat box, and Patil will read it out for the speakers to answer the questions and clarify the points. So having made this little elaborate summing up, I not only thank the speakers and the chairpersons, I also thank the local team, the organizing team from the University, the host institute, Dr. Patil and his colleagues who have done a wonderful job of organizing the whole thing, very meticulously done. And I have, of course, a lot of things to say tomorrow, so I thank them again. Dr. Patil, you can take over and make.
Yeah, thank you, sir. Thanks a lot. So thank you for nice sum up of the entire day sessions. Now we are having a dinner break in India here, and that break will be for 30 minutes. Already it is 20:38 here, so we're just having a break of 22 minutes now, and exactly at 9 p.m. we'll be starting with the poster presentation competition. We had got some participations from different places across India. Those posters were sent to juries for initial screening, and we have shortlisted 6 poster presentations for actual online presentation and questioning. Okay. So that event we are going to start exactly at 9 p.m. after 21 minutes. So let's come here together after 21 minutes. Please join to the same link, and YouTube also will be same link. Okay. Thanks a lot. I'm now closing the session, and we'll be meeting again at 9 p.m. Yeah. Thanks a lot. Thank you.
Very good evening, everyone. So, Dr. Christian, are you there? Hello, Dr. Christian. Sirians, am I audible?
Yes, sir. You are audible, sir.
Okay, okay.
Yes, sir.
We are just waiting for 1 or 2 minutes, then we will start.
Okay, sir.
Okay, sir.
Okay, 2 minutes. So, very good evening, everyone. Dr. Christian, are you there?
Hello, are you waiting for me?
Yes, yes, sir. Can we start, sir?
Yes, perfect. Thank you very much for your invitation to participate to the jury. I'm very honored and happy to be with you and all the attendees of this meeting.
Thank you so much, sir. Thank you so much for joining. So we have total 5 participants who are going to present the poster. So our first participant is Sirians, who is going to present. So participants, total duration is 10 minutes, including question and answers. So please be specific. Please, Sirians, share your screen and start your presentation.
Yes, sir. Yes, sir. Can you see my screen?
Yes, not yet. Just wait. Yes, yes, Sirians, go ahead.
Yes.
I hope, Dr. Christian, is it visible to you?
Yes.
Okay.
Yes,
okay.
yes, yes.
Thank you.
Okay. So shall I start?
Yes, go ahead.
Yeah. So good evening, everyone. I'm Shriyan Srivastav, and I'm really excited and glad to be here at SAAE, 4th annual conference of SAAE. And today, I would be presenting my work in this poster presentation, wherein I would like to tell what I did. And the title of my work that I did is in silico screening of compounds from plant and marine sources for inhibition of MK2 kinase. I have done this project under the supervision of Dr. Rakesh Kumar Singh from the Department of Pharmacology and Toxicology, NIFOR Raebareli. So this is a virtual. This is a experiment that was alternative to the regular animal models, and so I tried to screen the compounds virtually using different methods. So talking about the abstract of the work that I did.
I would like to talk about mitogen activated protein kinase. These are one of the many intracellular signaling cascades that respond to various types of extracellular stimuli. The MAP kinase pathway get activated by phosphorylation at different levels, and MK2, basically talking specifically about MK2, it is a kinase that is downstream to P38 MAP kinase and is activated by phosphorylation with ATP. The role of MK2 has been investigated in various types of chronic inflammatory diseases, and the investigation is also going on for COVID. The compound ATI-450 is a potential target that, and MK2 is being investigated in COVID pathology as well. Until now, there have not been any. There have been a lot of investigation for small molecule inhibitors. We have tried the researchers have tried to develop small molecule inhibitors, but the area of natural compounds have not been explored for MK2 kinase. Therefore, I use different types of in silico softwares that are freely available online to screen the potential compounds, and I got some really good results that could be utilized for further experimentation, thereby reducing the use of animals. Talking about the highlights of the protein that that I used, basically the role of MK2 lies in that MK2 regulates the pro-inflammatory cytokines. The ATP binding site of MK2 is very deep and narrow. Only small planar molecules can bind to the ATP binding site, and the crystal structure of the MK2 protein that I used was resolved at 3.20 angstrom. Talking about the methods of the work that I did, basically what I did initially was I took a target protein structure from protein data bank, that was 3R30. This is a protein chain wherein the ATP binding site was present of the MK2 kinase. Further, I use different types of libraries that are present freely to select marine and plant compounds using from literature review and Tanimoto coefficient search. Then I prepared the ligands, and I followed the procedure and performed docking studies using Autodock Vina 4.2 at the MK2 binding site. This docking was site selective, and the grid that I used was 20 units XYZ. Further, I analyzed the molecules for their ADME properties and the target prediction, and I got some really great results. The scheme to explore potential compounds. So initially, we reviewed the natural compounds that have been previously investigated for kinases, based on the hypothesis that kinases share some degree of homology. Further, we docked these compounds and we ranked these compounds, the compounds that had good ranking were further projected to target prediction and ADME properties. Additionally, the compounds showing good binding affinity were also subjected to similarity search through Tanimoto coefficient search using IMPP80 database. Whatever softwares I used were totally available for free, and anyone can explore them. The results that I obtained from this study was that when I saw the binding affinities of the internal ligand CD2 that was bound to this protein 3R30, it came out to be minus 8.9 kcal, which is a very good binding affinity. Further, I used the tool compound PF3644022, that is a commercially available MAP MK2 inhibitor, and the binding energy came out to be minus 9.3, which was which actually validated that the binding and the interactions were possible. I further screened 166 compounds over a period of 4 or 5 months, and I shortlisted 4 compounds, that is dracorubine, crinaceatin K252C, and azotoxin. The binding energy of these were comparable as that to the internal ligand and were in the range of 11 to 9. When I saw these, when I visualized the interactions of the compounds, of the tool compound, of the internal ligand, and of the natural compounds that I docked, what I saw that the type of interactions were different, but the amino acids which were interacting with this scaffold were similar, and most of the most favorable bonds, the most favorable amino acids that showed interaction with the ligand or the natural compound that we were investigating were methionine, cysteine at 140 amino acid site, so methionine at 138, threonine at 206, glycine at 93, lysine at 70, and so on. There were a lot of interactions, and thus we moved further with the ADME prediction of these 4 compounds and target prediction of these 4 compounds, upon which we realized that the probability of these compounds to target kinase is there, and thus I would actually like to conclude from this study that naturally derived compounds with diverse scaffolds have shown good binding affinity with MK2 kinase, and the results suggest that compound azotoxin, crinaceatin, and K252C can be used for further evaluation. Summarizing the result, I would like to just give me a moment, sir. I'd like to zoom the table. Summarizing the result, the compound crinaceatin had a great binding affinity of -10. The target prediction towards kinases was 40%. The ADM and drug likeness was favorable, and the same was for K252C and azotoxin. So what I wanted to conclude with this is that it is a really great study. I did not spend a lot of money. It was very commercially feasible, and it was accessible to everyone. Lastly, I would like to thank Dr. Rakesh Singh, Dr. Anup Kumar, Department of Pharmaceuticals, Ministry of Chemical Fertilizers, and NIBOR Borelli for all the support that they have provided in completion of this study. Thank you.
Thank you, Sriyansh. Thank you very much. Now over to Dr. Christian.
Thank you very much for the presentation. Very clear. You have done a huge work, and the progression is very clear. It's what I like. I have a question, maybe.
Yes, sir.
If I can.
Yes, sir. Yes, sir, you can. Yeah.
Yes, sir. Yes.
The three products that you chemical that you selected, are they already used for some drug, or are they already on the market in some product?
Sir, from my knowledge, they are being investigated. They're not available in the market as of now. They're being investigated. They're in the preclinical and clinical development stages.
Okay, so it's a new application for the future. You have to find the good partnership to go further in the development.
Yes, sir. Yes, sir, definitely. So, in fact, I'm looking forward to different types of studies as well right now on this, and we would be moving forward with this. My supervisor is working on this, and I'm positive that we would be, give out something really fruitful from this.
Perfect. Thank you.
Thank you very much, sir. So now we are moving to next candidate. Next candidate is Rakesh.
Yes, yes, sir.
Rakesh, Sriyansh, you stop sharing screen. Rakesh, you share your screen.
Yes, sir. I am sharing screen, sir. Sir, my screen is visible, sir?
Yes, it's visible to me. I hope it's visible to Dr. Christian also.
Yes, it is.
Yes, sir.
Okay, Rakesh, go ahead.
Yes, sir. Thank you, sir. This is good evening to everyone. This is P. Rakesh Kumar. I am going to discuss about the study Qstr modeling for predicting reproductive and hepatotoxicity. My supervisor is Ashok Kumar Datsalia, sir. Department of Regulatory Toxicology, NIBOR Borelli, India. Abstract of the study is determining the toxicity of chemical compounds is one of the essential steps in drug design. So far, animal models have been used for toxicity testing and are constrained by time, ethical issues, and financial burden. In this study, Qstr model was performed to develop a model for predicting reproductive and hepatotoxicity of unknown compounds. Target proteins used in this study are BRAF gene, also known as proto-oncogene, estrogen gene, estrogen receptor. Objectives of the study were to calculate the molecular descriptors for selected compounds, to select an appropriate genetic algorithm, to develop the best fit model for predicting reproductive and hepatotoxicity, to perform docking studies of the known and query compounds with validated target proteins. Coming to the methodology of the study, in the first step, data curation, we have collected IC50 values for the estrogen receptor and BRAF gene from different online databases like PubChem, Chembel, DrugBank, Toxnet. Then after, we have calculated molecular descriptors from chemical structures for those chemical structures with the help of Paddle descriptor software, eDragon software, ChemDes software. We have calculated 1544 molecular descriptors for both the models. Then after, statistical mapping of descriptors to a toxic endpoint is done with the help of DTC QSCR software. Then model validation, we have done internal validation and external validation. Next, coming to the applicability domain testing. Finally, model is developed. Now it will be used for testing query compounds. Results of the study was, firstly, 2D Qstr model and validation results for hepatotoxicity. We developed an equation for correlating the biological response with the descriptor. In this equation, there are 3 descriptors we have selected. Those 3 descriptors, the descriptor with negative sign indicates the negative contribution in the model development. Positive sign indicates the descriptor having more contribution in the model development. We have calculated internal validation parameters, leave one out result, external validation parameters. All these parameters are get passed according to the OECD validation criteria. Based upon the above results, model quality based on these results were good. 2D Qstr model and validation results for reproductive toxicity were, again, we have developed an equation. In this equation also 3 descriptors were there. These 3 descriptors are again with negative and positive positive signs that they are contributing negative and positive contribution in the model. We have calculated internal validation parameters, leave one out result, external validation parameters without scaling, after scaling. All these results are passed according to the OECD validation criteria. Model quality based on MAE based criteria was good. We also performed the external validation for this model. External validation we performed by taking the compounds which are not involved in this model development. In this model, for external validation, there is no systemic error present in both toxicities, and prediction quality is good for both models. The mean absolute error after removing 5% test set objects with high residual values was found to be 0.06513 and 0.02364 for hepatotoxicity and reproductive toxicity respectively, which is surely acceptable. Error-based matrices we have followed for both the models, and for both the reproductive and hepatotoxic model, the model prediction quality was obtained was good. We determined response plot for those models. There is a linear relationship between the predicted and observed values were good in both models. Correlation coefficient of R square is 0.94 and 0.996 for hepatotoxicity and reproductive toxicity respectively. Cross-validated regression coefficient of Q square is 0.93 and 0.995 for hepatotoxicity and reproductive toxicity respectively. This is the response plot for hepatotoxic model. This is the response plot for reproductive toxic model. We also developed residual plots by subtracting the observed value minus predicted value. Now coming to the applicability domain. Applicability domain was defined as a structural outlier about a reliability about the chemical moiety. In this applicability domain for reproductive toxicity, only one compound in training set was considered as a structural outlier. Remaining all compounds in test set and external test set all are inside the domain. While coming to the hepatotoxic model, all are inside domain. All in training set, test set and external test set, all compounds are inside domain. Now coming to the query compounds we have taken, 29 query compounds we have taken, and we have evaluated the applicability domain status for those 29 query compounds. Some compounds are outside the applicability domain, some are inside the applicability domain. The compounds which are outside the applicability domain are considered to be non-toxic, and compounds which are considered to be inside the applicability domain are considered to be toxic. We also performed docking studies for both the known toxicants and query compounds. Here we represented only the known toxicant docking studies. These two compounds are hepatotoxic, known hepatotoxicants. Their target was BRAF gene, and this image shows that there is a binding affinity between BRAF gene and ligand. Docking for reprotoxic model results are interpreted based on binding energy. These two compounds are known reproductive toxicants. Their target was estrogen receptor. Here this image shows that there is a binding affinity, a strong binding affinity between estrogen receptor and toxicant. Conclusion of this study was, we have in reproductive toxic model, we have we have selected 29 query compounds. In those 29 query compounds, 25 are inside the applicability domain, one is outside the applicability domain, one most active. One most active in the sense, based upon the docking studies, that one molecule have higher binding affinity with the target. In the hepatotoxic model, we selected 29 query compounds, 11 are inside the applicability domain, 18 are outside the applicability domain, 2 are most active. These two compounds are, according to the docking studies, these two compounds are mostly binding, strongly binding affinity with the target, that is BRAF gene. By this in silico investigation, we have successfully developed Qstr model for hepatotoxicity and reproductive toxicity. The statistical matrices of the descriptors showed that the model is robust and statistically significant. Hence, from the study, we concluded that kynurenolic acid is non-toxic and its analogs are predicted as toxic due to structural change at the acid group. Finally, the authors thank the Department of Pharmaceuticals, Ministry of Chemicals and Fertilizers, Government of India, and Director NIPER for support and resources. And sincerely thanks for giving me this opportunity to present my study for Dr. Anup Kumar sir and SAAE conference. Thank you very much.
Thank you, Rakesh. Thank you very much.
Thank you, sir.
Now over to you, Dr. Christian.
Thank you very much. Very clear. A little bit far from my expertise, the in silico, but it's very promising for the future to be able to replace the animal. I just have a question. Maybe I didn't catch it. You did. You said it. But if I well understood, you selected some unknown compounds, and you developed a predictive model. And after you tested your selected compound in your predictive model, my question is, to validate your models, how many compounds have you got used with? I suppose for all these compounds, you have already the in vivo data. So it's the size of the validation set which is my question, in fact.
For the query compounds, there are no in vivo data. These are, those are before synthesized, we are predicting their toxicity. But for these unknown compounds, with the unknown compounds, we have developed the model. For these unknown compounds, we have in vivo and in vitro data, sir.
Okay, so how many compounds, I'm sorry, but how many compounds you use with in vivo data?
31 molecules for hepatotoxic model and 60 compounds for reproductive toxic model we are used, sir.
Okay. Thanks.
Yes.
Thanks very much.
Thank you, sir.
Thank you very much. So now we have next candidate, Meenakshi. Meenakshi, are you there? Rakesh, you stop sharing screen. Meenakshi,
yes, sir.
Yes, Meenakshi, share your screen.
So I'm unable to share the screen because it is mentioning that host disabled attendee screen sharing.
Just give me 1 minute. Yes, now I have made you host. Meenakshi, now share your screen.
Yes, sir.
Are you able to share?
Yes, sir.
Yes, share yours. Go ahead.
So is the screen visible?
Yes, it's visible to me. I hope it's visible
Yes.
to Dr. Christian also.
Exactly.
Yes, Meenakshi, go ahead.
All right. A very good evening to all the SAAE members, organizers, and all the attendees. My name is Meenakshi Gupta, and I am a third-year PhD scholar from Amity Institute of Pharmacy. I'm carrying out my research work under the supervision of Dr. Mariam Sarbat. Her lab mainly focuses on the anti-cancer and neuroprotective activity of herbal molecules. We have done the in silico screening of plant-derived natural compounds from daba and curcum to explore their anti-axis activity. So, hepatocellular carcinoma, as we all know, is the major cause of cancer mortality throughout the world. Treatment with sorafenib, which is considered the first-line management, prolongs the survival of patients by only 8 to 11 months and has various complexities associated with it. So we come up with the idea of evaluating the anti-cancer potential of a plant-based Unani formulation, Dava ul Kurkum, and it is generally prescribed by Unani physicians for the patients having liver and gallbladder enlargement. It has components from 7 plants, namely cinnamon cassia, having a major chemical constituent, cinnamaldehyde and B-coumaric acid. The second biological compound is cinnamon zelanicum, having cinnamaldehyde. The third one is Symphobogan druangusa, having citral as an active constituent. The fourth one is Crocus sativus, possessing crocin, quercetin, safranal, picrocrocin, camphophyrol as an important constituent. The fifth one is Commiphora vitae, possessing glucostriol as an active constituent. Sixth one is Nardistachys jetamansi, which has zetamanson and isovaleric acid. The seventh one is Sosoria lapa, which has dihydrocostus lactone as an active constituent. So the objective of the study was to analyze the anti-cancer activity of Dava ul Kurkum, first as in using the in vitro approach, and then to gain the functional insight, we used in silico studies of the natural compounds which are present in Dava ul Kurkum. So we followed the methodology. First, for the in vitro screening of Dava ul Kurkum, we performed entity assay, scratch assay, colony formation assay, and cell cycle analysis. And then for the in silico screening, we have used molecular docking and MD simulation studies. Moving to the results, when the cells were treated like both malignant and non-malignant cells, we have used, for malignant, we have used HepG2, Hep3b and HUH7, and for non-malignant cells, we have used HEK293 and primary hepatocytes for comparison. So when these cell lines were subjected to different concentrations of Dava ul Kurkum for 24 hours, and we quantified the result based upon the forms information, we observed that Dava ul Kurkum was able to significantly reduce the cell viability of malignant cell, but it was very minimally affecting the viability of normal cells, like in particular HEK293 and primary hepatocytes. So we considered that the data is of interest as a drug is able to inhibit the viability or the activity of cancer cells, but is very minimally affecting the viability of the normal cells. Next, we observed the effect of Dava ul Kurkum on the wound healing assay of the Hep3b cells. Why Hep3b cells we have chosen? Because we have gained a very minimal IC50, or the least IC50 was observed in the Hep3b cells. So we carried out further experimentation on Hep3b cells. So we observed the effect of Dava ul Kurkum on the wound healing property of Hep3b cells, and we observed a significant reduction in the wound healing capacity of the cells which were treated with Dava ul Kurkum as compared to the cells which were treated with the vehicle, that was DMSO. So to further investigate the anti-cancer potential of Dava ul Kurkum on Hep3b, colony formation assay was carried out. Generally, cancer cells have the tendency to grow in colonies, and they contact with their neighboring cells, and when they lose this this connection with the adjacent cells, this leads to the death of these cells. So the similar pattern was observed by the colony formation assay when the cells were treated with IC30, IC40 and IC50 of Dava ul Kurkum. We observed a dose-dependent decrease in the colony formation assay of Dava ul Kurkum. Next, we have done the antioxidant activity of Dava ul Kurkum, and we determined this activity using DPPH assay and FRAP assay. In DPPH assay, we observed that Dava ul Kurkum has a similar effect as that of ascorbic acid, which is taken as positive control at a concentration of 30 microgram per ml, and as shown in the graph, it was observed that there is a dose-dependent antioxidant activity of Dava ul Kurkum. The similar results were obtained from the FRAP assay. Then the cell cycle analysis was done to determine in which in which stage of the cell cycle is arrested after treatment with dawarcurcum, and we observed a significant increase in the cells which were present in the G2M phase. A significant decrease in the cells present in G0, G1 phase was observed, and a significant increase was observed in the cells which were in G2M phase. So we can deduce that the cell was arrested in the cell cycle was arrested in G2M phase. Now, to gain further insight of the activity of dawarcurcum, we carried out in silico studies, and for that we docked 10 active constituents present in the 7 herbal components of dawarcurcum, and after the docking, the preliminary in vitro screening and the admit profiling, and even the results were compared with the standard drugs, we observed that out of the 10 active constituents, Z-glucosterone was showing the best activity and was binding very well with the 4 selected proteins. So, as shown in the figures, Z-glucosterone showed hydrogen bonding with asparagine 6 and 74 with azarine gene, glutamine 112 and serine 92 binded with aminoacylase 1. Lysine 427 was binding with RB. Lysine 427 of RB1 gene was binding with glucosterone, showing hydrogen bonding, and there was a van der Waals interaction with the calreticulin protein. The results were validated by MD simulation studies as well, and we observed that for both the studies, like for the docking studies and for the MD simulation studies, there was a similar scaffold in which our molecule Z-glucosterone was binding with all the 4 proteins. So this table shows the binding affinity, and we observed that the binding energy was all in negative, and for RB1 protein, we observe a binding energy of negative 11. For azarine, it was negative 17, and for aminoacylase, it was negative 100. For calreticulin, it was negative 8. Then the RMST deviation plot was observed. And for all the 4 selected proteins, we observed that the RMST plot showed a value less than 0.5 for all the proteins, showing a stable binding in the scaffold of the protein. So with all the findings, we can conclude that dawarcurcum could be a novel candidate against HCC, and its component, particularly Z-glucosterone, as per our in silico study, can be explored further for the drug development and lead development. Thank you, everyone.
Thank you, Meenakshi. Thank you so much. So now over to you, Dr. Christian.
Thank you very much. Very interesting to mix in silico approach and in vitro approach. If I well understand, you have identified some compounds that could be particularly active, some compounds extract from the dawarcurcum plant.
Yes.
What will be the next step for you now? Because if you are convinced that this compound are very effective, maybe a future drug development is envisaged?
Yes, sir. Next, we will go for the in vitro studies of Z-glucosterone, and also when we will be satisfied with that, then we will move for the in vivo studies of the same.
Okay, thank you. Very interesting.
Thank you so much, sir. Thank you.
Thank you so much, Meenakshi. Now, next candidate is Aarti. So you can stop sharing.
Yes.
Aarti, are you there?
Yes, sir. Am I audible?
Yes. So you can share your screen. Yes, it's visible to me. I hope it's visible to Dr. Christian also.
Yes.
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 So
Can I start?
yes, Aarti, go ahead.
Good evening, everyone. I'm Aarti Ashok, Junior Research Fellow at Sri Chitratirunal Institute for Medical Sciences and Technology, working under the guidance of Dr. P.V. Mohan. The title of my poster is Organoid, a novel approach for predicting cytotoxicity. So, organoid technology presents an alternative method for predicting nanoparticle toxicity assessment, and there's a promising technology that links the existing preclinical models with human physiological studies. The toxicity of nanoparticles is tested in animal and cell culture models to predict its effects in the human system. Due to the ethical concerns and species variation, animal models are reduced and cell culture models have been considered to provide cellular responses for decades. However, these models still lack the efficiency to the cyclic physiological response.
Aarti, your voice is not clear. It's breaking.
So sorry, maybe my connection is not stable.
Now it's okay. Go ahead.
Hence, the organoid technology is predicted to bridge the gap between in vitro and in vivo environments. So in this present study, the in vitro responses of cysteine-capped zinc oxide nanoparticles on A549 cells was investigated in conventional cell culture systems and a microfluidic device under a physiologically relevant flow rate. So the methodology involved four parts mainly: the synthesis of nanoparticles, the characterization of the nanoparticles, and the conventional cell culture assays and the microfluidic assays. So the method for comparing the cellular responses of the nanoparticles treated cells using conventional cell culture system and microfluidic cell culture system was the main aim of the study. So in the conventional in vitro methods, we have used various assays like the MTT assay, lysosomal activity by neutral red uptake assay, LDH assay, intracellular ROS production by DCFH-DA assay, nitric oxide generation by grease test, etc. Then the staining methods like the rhodamin colloidin for cytoskeletal stability, acridin assay for lysosomal membrane studies, JC1 staining for mitochondrial membrane, and adaptive staining for nuclear condensation were performed. So from the conventional assays, we could conclude that the concentration 160 microgram per ml was toxic to the cells. Hence, this concentration was used in the microfluidic cell culture to compare it with the normal static cultures. So in the microfluidic cell culture, the calcium PI was done to estimate the live and dead cells in the device. Lactose dehydrogenase release assay was done. Then the nitric oxide release was also measured. Then moving on to the staining techniques, we also perform rhodamin colloidin staining and lysosomal membrane integrity by activating orange on the chip. So moving on to the results, the microfluidic organoid device was designed to investigate the cellular uptake and toxicity of cysteine-coated zinc oxide nanoparticles under dynamic and static conditions. So as you can see here, we have performed various assays to compare the cellular responses with both conventional and organoid approach. The cysteine-coated zinc oxide nanoparticles was performed in both static and dynamic conditions. The morphology changes in the static and dynamic conditions and before the particle treatments were visualized. The cysteine-coated zinc oxide nanoparticles treatment in static conditions demonstrated more cell death than dynamic conditions, which is also visible in the live death staining by calcium PI. The acridin orange assay to visualize the variation in the lysosomal integrity exhibited a subtle change in the static nanoparticle treated cultures as compared to the dynamic nanoparticle treatment conditions. Static nanoparticle treated demonstrated more damage to the lysosomes. The rhodamin tappy staining of the cells in the device, the rhodamin tappy also showed. Hello, am I audible, sir?
Aarti, your voice is breaking.
Sorry. So the rhodamin tappy staining of the cells in the device ensured a proper attachment of the cells on the walls of the microfluidic device. The static treatment of the cysteine-coated cellular nanoparticles resulted in more damage to the cytoskeletal integrity than the dynamic conditions. However, under dynamic condition, the particle treatment resulted in decreased LDH release. So, the microfluidic study for the toxicity examination of cysteine-coated nanoparticles demonstrated that the toxicity induced by nanoparticles depends on the residence time, local concentration, and perfusion. The dynamic condition can reduce the residence time of the nanoparticle due to toxicity. The flow-integrated nanoparticles induced in vitro can also provide more relevant cell responses. In static conditions, the nanoparticle can settle upon the cell, thereby increasing the local concentration, which may lead to increased cell death. Therefore, it is necessary to consider the residence time of the nanoparticle, the local concentration, and the flow conditions at the target cell while predicting the toxicity associated with the nanoparticle exposure. Hence, to conclude, the organoid chip technology can be a good alternative for animal experiments. So, thank you.
Okay, thank you, Aarti.
Thank you. I'm really sorry, but the sound was very bad.
Yes.
It's a pity because your work is very interesting, and especially the effect of dynamic or static condition are interesting. I didn't catch up all what you said, but in one word, can you just remind me what is the best solution, static or dynamic, and why it is the best?
Pardon, sir? It's not clear.
I'm sorry.
I think Aarti has some network issue, sir.
Hello?
Okay.
Yes, Aarti?
Yes, sir, I'm here. My net connection is not stable.
You don't hear me? My question was just confirm, say me what is the best condition, static or dynamic?
Yes, sir,
and
dynamic
why
conditions.
Dynamic, and
Of
why
course,
it is
dynamic conditions, because in static conditions, the nanoparticles tend to settle down and give more stress to the cells. But in the dynamic conditions, as it is moving continuously, there is no chance of settling of these particles, and the cells would grow more efficiently.
Thank you. I didn't hear all of your answer, but thank you, and it confirms the added value of microphysical system in dynamic condition, and thanks a lot, and sorry for the bad connection for you.
Yeah.
Thank you, sir.
Thank you, Aarti. So now we have next candidate. Nidhi, are you there? Aarti, you can stop share your screen. Nidhi, are you there?
Yes, sir.
Yes, you share your screen.
Okay, sir.
Aarti, you stop sharing. Okay, yes.
Is it visible, sir?
Yes. It will take a little bit time. Just wait. Yes, it's visible to me, and I hope it's visible to Dr. Christian also.
Exactly.
Yes. Go ahead.
Okay. Good evening to one and all present here. I am Nidhi Sharma, second year PhD scholar from Amity Institute of Pharmacy, Amity University, Noida. I am here to present my research work entitled Protective Role of Camphophyrol Altering ER Stress Signaling in Hepatocellular Carcinoma. I am carrying out this research work under the kind guidance of Dr. Mariam Sarwat and Professor Subhrajit Biswas. Now, talking about the background of the study, hepatocellular carcinoma is the most lethal primary liver cancer that accounts for about 80 to 90% of cancer-related mortality worldwide. Hepatitis B virus, hepatitis C virus, aflatoxin-contaminated food, alcohol abuse, and non-alcoholic fatty liver diseases, etc. are some of the risk factors that contribute directly and indirectly in the pathogenesis of HCC. Typically, HCC is usually diagnosed at an advanced stage, and many patients with advanced HCC are not eligible for curative therapies. Thus, the identification of a novel therapeutic approach for treating HCC is urgently needed. Camphophyrol is a dietary aglycone flavonoid that is present in numerous plants, fruits, and vegetables such as aloe vera, Euphorbia pekinensis, broccoli, apple, and grapes, etc. It is known to selectively kill the tumor cells without affecting the normal cells. However, the killing effect of camphophyrol on HCC and its underlying mechanism are still not clear. Camphophyrol is well known for the diverse pharmacological activities, including antioxidant, anti-inflammatory, cardioprotective, neuroprotective, and anti-tumor, etc. Camphophyrol is explored against various pathways such as mTOR, APK signaling pathways, etc. But is in cancer, but it is not yet explored against ER stress signaling pathway. So in this study, we are trying to target camphophyrol against HCC via ER stress signaling pathway. The objective of my study is to compare anti-cancer potential of the saffron flower constituents, that is saffron petals, stamen, and stigma. We have completed this objective. In this objective, with the help of MTT assay, we have basically evaluated the anti-cancer potential of the three constituents of the saffron flower, and the results demonstrated that among all the constituents tested, saffron petal has shown better cytotoxic activity. So we have selected saffron petals for the use in subsequent study and eliminated the remaining constituents. Next, we have evaluated the anti-cancer potential of the secondary metabolites that are present in the saffron petals, and the secondary metabolites which we have tested were crocin and camphophyrol. In between both the metabolites tested, camphophyrol has shown better cytotoxic activity. So the second objective is to study the principal natural component of saffron petal, that is camphophyrol, against hepatocellular carcinoma. The last objective is to identify camphophyrol targeting molecule via computational analysis and their validation through in vitro studies. Now moving on to the methodology of the study, my entire research work is divided into two major categories: in silico studies and in vitro studies. Under in vitro studies, we have performed MTT assay, scratch assay, gene expression analysis of several apoptotic and ER stress related proteins. In computational studies, we have targeted camphophyrol with several ER stress related proteins. Now moving on to the results, we initially investigated the effect of camphophyrol on the cell viability of different cell lines. In this, we have in this liver cancer cells as well as normal liver cells that are our primary hepatocytes were exposed to the different concentration of camphophyrol, and the results demonstrated that camphophyrol significantly reduced the cell viability of the liver cancer cells while exerting no harmful effect or cytotoxic effect on the cell viability of primary hepatocytes. Among all the three cancer cell lines tested, Hep3b has shown greatest reduction in its cell viability. So on the basis of this, it can be concluded that Hep3b cell lines are more sensitive to the camphophyrol treatment. So we have selected Hep3b cell lines for use for subsequent subsequent experiment to determine its potential mechanism of action in the treatment of hepatocellular carcinoma. Next, we evaluated the effect of camphophyrol on the migration of liver cancer cells. Cell migration were assessed with the help of scratch assay or wound healing assay to determine the anti-migratory effect of camphophyrol on liver cancer cells. So the results demonstrated that camphophyrol significantly inhibited the migration of liver cancer cells. Now coming to the results of computational studies, in computational studies, we have selected several proteins that plays a vital role in ER stress related pathway, and among all the selected proteins, camphophyrol has shown wonderful binding affinity with PERK and IRE1 alpha. PERK and IRE1 alpha are the proteins that plays a major role in endoplasmic reticulum stress pathway and causes cell death either by apoptosis or autophagy. So the binding affinity we observed in case of PERK was minus 9.1 kilocalories per mole, and in case of IRE1 alpha, the binding affinity was found to be minus 7.2 kilocalories per mole. Next, we evaluated the gene expression analysis of camphophyrol, the shortlisted ER stress related protein. These are the proteins which we got from the computational studies. PERK and IRE1 alpha are the proteins that causes cell death by apoptosis or autophagy mechanism. So in the conditions like HCC, the expression level of PERK and IRU1 alpha got downregulated. But when the liver cancer cells were treated with camptoferol, the results demonstrated that camptoferol significantly upregulated the expression level of PERK and IRU1 alpha in a time-dependent manner. Next, we have performed the gene expression analysis of camptoferol with the several apoptotic genes. So, caspase-3 is basically an executional protein that plays a vital role in carrying out the apoptotic process, whereas BCL2 is an anti-apoptotic protein that that plays a major role in inhibiting the apoptotic process. So, when the cell lines were treated with camptoferol, the results demonstrated that the level of caspase-3 were upregulated significantly in a time-dependent manner, whereas the expression level of BCL2 were downregulated significantly in a time-dependent manner. So on the basis of this finding, it can be concluded that camptoferol may induce hepatocellular carcinomic cell death via ER stress-related pathway. We have also filed a patent in the second year, having a patent number this, and also we have published a paper entitled Antioxidant Role of Camptoferol in the Prevention of Hepatocellular Carcinoma, but published in an antioxidant journal. Thank you so much for your valuable time and attention.
Thank you very much, Nidhi. A great job also with patent and publication, so congratulations. I have a question, maybe, because I am not an expert and naive, but it's about the in vitro assay you performed. When I have a look to your poster, if I well understood, it's a test on the capacity of the cells to migrate.
Yes, sir.
And in the control at 0, 0, and 24 hours, is there a migration or not in this control? There is no migration normally in the control, or there is migration of the cells?
Also in the control group, we found that the cells were migrated from 0 hour to 24 hours, but when the cells were treated with camptoferol, the migration of the liver cancer cells were inhibited.
Okay, it's not clear for me the migration, the difference, because in fact, in 2D, you're doing a scratch on the 2D cell culture, and after
yes,
you follow the disappearance of the scratch.
Yes, sir.
But between 0 and 24 hours, I
Maybe
don't
the
see.
pictures are not very clear.
Okay. Okay.
Okay, sir.
Thank you very much.
Thank you so much.
Yeah, thanks a lot, Dr. Christian. So here we are finishing all the 6 candidates who were shortlisted by juries, 5 candidates who were just shortlisted by juries. Thanks, Dr. Christian. And due to time constraint and little anxiety about starting at the specific time, we left behind introducing our jury, Dr. Christian Pellivaisian. I'm doing that thing now. Dr. Christian Pellivaisian is a biologist and toxicologist with expertise in the in vitro safety and efficacy testing based on 3D reconstructed human tissues. As scientific director of Episkin, he has been involved in the development of reconstructed human tissue and in vitro methods to assess the safety of chemicals, cosmetics, drugs, and medical devices. Involved for several years in biocompatibility of medical devices, he is chairman of the French APNOR Commission for biocompatibility of medical devices and convener of WG8 skin irritation and sensitization of ISO Technical Committee 194 for biocompatibility of medical devices. Dr. Christian had been associated with all the stalwarts who are now office bearers of SAAE-INDIA, and she has been a supportive force for SAAE-INDIA and the activities of SAAE-INDIA since long. I would like to request Dr. Akbar Shah sir to say a few words about Dr. Christian Pellivaisian, who has sponsored the awards for poster as well as oral representations. And Dr. Akbar Shah will be elaborating a little more about please, sir, please.
So, well, as I was telling you during the sum up session after the plenary, Dr. Toloi Christian is known to me for more than a decade. We meet in many conferences and other activities, and a lot of intimacy was developed because of a lot of prevalent compatibility. And particularly, Dr. Christian was interested in like giving training to young minds, young students, so that they would practice the alternative methods. You know, Dr. Christian comes from the company which is the Episkin. Okay, Episkin company is in France, and the company makes the reconstructed human epidermis, human tissues, which is actually primary cells which are laid on matrix, and the cells are alive, and they could be kept alive for a number of days, defined number of days, and they could be used for testing of like cosmetics or other chemicals for irritation and corrosion, as you would do in the trace test, skin test on rabbit. So the reconstructed human epidermis is an OECD approved alternative to trace eye test, trace skin test, and of course there is another model, reconstructed model for cornea, which can be used in the place of trace eye test. So Christian is the scientific director of that company, the Episkin, which is approved. The product is approved by the OECD. So with that tremendous background and enthusiasm, Christian offered to be associated with trainings in India. He has a lot of faith in university-industry collaboration. That is a very fancy word for him to be using in many conferences, and he used to invite me to speak about the outcome of the collaboration between Episkin and the Mahatma Gandhi Development Center, about which I was associated earlier, and the Society for Alternative Standard Experiments now. So, very nice person, wonderful, very knowledgeable person about alternatives, and a lot of biochemical expertise in biochemical investigations. One of the a very good friend, more of all these things, a good friend of mine. So it is very nice that Christian didn't stop with volunteering to train people, but he even volunteered. He was so impressed by the functioning of our society. He volunteered to bring money from his company, the Episkin, and its parent company, the L'Oreal, to bring money to award prizes for oral and poster at the annual conferences of the Society for Alternatives. So he is virtually the host of this session. That is, he means me in the company, the Episkin, and the L'Oreal are the host of this session, oral presentation and poster presentation. Today is poster, and tomorrow we have oral. So, I mean, given to me, I go on speaking. I should stop. So the association is so strong.
Yeah.
The association is strong. I can't control. Speaking about Thomas, speaking about Christian, I can do endlessly, right? So, but then it will be monotonous for others. So I stop here. It is very nice of Dr. Christian to have accepted our invitation to be a jury for his hosted session, the poster award presentation. Thank you, Christian. And
thank you very much, Professor Akbar Shah.
Patil, you could write.
Okay, thanks a lot, Dr. Akbar Shah, sir. And thanks a lot, Dr. Christian. Here we are now at the end of this poster session. I would like to request Dr. Christian to say a few words about the contestants and their presentations, so that we can wind up this particular session, and we'll be then concluding the entire day's sessions. Dr. Christian, please.
Thank you. we wait, I suppose, to discuss after to give the name of the award. We don't say it we will discuss after, okay? Thank you very much for inviting me. Thank you very much for the introduction by the organizer to this session. I think that this session is very important. For me, replacing the animal in toxicology will take time, and it's a question not only of scientific technology, but it's also a question of humanity and human, and having more and more people aware about the challenges, able to generate data, able to convince also the regulators. So supporting the Society for Alternative to Animal Experiment for me and my company, it's a commitment, and supporting also the young researchers in this direction is very, very important. So that's why I'm happy, and very happy also to see the quality of the speakers and of the work. So it will be not easy to select the winner, but I think that with discussion with my colleague, we will make the best choice for this.
Yeah, sure. Thanks a lot, Christian. Dr. Christian, we are really thankful to you for sparing a lot of time for this session and then tomorrow's session also, and for evaluating the submissions through emails that were sent to you. So actual winners will be declaring in tomorrow's session after the finishing of oral presentations. For both the sessions, we'll be declaring the award winners tomorrow itself. Yeah.
Okay.
So we'll be welcoming you again tomorrow evening for this evaluation. Professor Aisuddin sir, would you like to say something at the end of this day? Sir, you have to unmute. Dr. Aisuddin sir, you have to unmute. No, you're not audible yet.
It's fine?
Yeah, now you're audible. That's fine.
Oh, okay. So after a long time, we are seeing Christian in virtual way. So so nice to see you. You little bit aged. Any problem? You are looking little bit more grays are there. Huh? Can you unmute your mic, please? Christian, Krish, can you unmute your mic, please?
Yes. Excuse me.
You are aging gracefully.
Pleasure to see you, Professor Aisuddin. Also.
Yeah, I was also so pleased. So very wonderful sessions we have today, and all went well. I think tomorrow also afternoon we are meeting.
Right.
So thank you, Christian, for being with us and also making generous contribution to the efforts of society. And you can see we are doing very honestly, as much as possible, with too much transparency. We are conducting society. So people like you are around. Thomas was there, his wife, and so many speakers. So many people, many students have joined. So I think partly we are now saying goodbye, good night for today.
Yeah, right, sir, right. So
Yeah.
here we are trying to conclude this session. I am thankful to everyone who was there since morning with us. We had all the SAAE life members.
I was today that I had lunch also from Dr. Patil.
So thanks a lot, sir, for joining the live session also. We had the president, vice president, executive committee members, live members of SAAE-INDIA with us, and Dr. Christian also. All the speakers, they really put as much efforts as possible to make this entire day a successful day for SAAE-INDIA's 4th annual conference. I'm thankful to each one of them on the behalf of DIPSARU, on the behalf of SAAE-INDIA also. So tomorrow again, we are going to meet at 2:30 p.m. as per Indian Standard Time. The link for joining the sessions will be same that has been shared with all. Even YouTube link will be the same. You will have to join through this. Means participants will be joining through the same YouTube channel that has been shared with them. So let's say good night to all. And this is where we are summing up and concluding this first day of 4th annual conference of SAAE-INDIA. Thanks a lot for joining. Thank you very much.
Thank you, Dr. Park. Thank you very much. Have a good night.
Yeah, good night. Good night.
Yeah, good night, sir. Good night.
Thanks for everything.
Yeah.
We meet tomorrow.
Yeah, sure, sir. Sure, we'll be meeting tomorrow. Bye. Bye, Dr. Krishnan.
Bye-bye.
Bye, sir. Bye. Yeah. So all contestants can leave now. We'll be meeting tomorrow again. Thank you.
Hello, excuse me, sir. May I ask something?
Yeah, please go ahead.
Sir, myself, Dr. Uzma Rafi. I am from Regional Institute of Education, Ajmer. Here I am working as the assistant professor in zoology. Sir, I just want to ask you that how can we register in this conference? Actually, I get this link from some of my friends send me this link. So I want to register myself in this conference. So is there any option for this?
Okay, see, I'm sending you a link.
Okay.
You can call me separately. Okay, you just write down my number here.
Okay.
It is 9823.
Just a second, sir.
Okay, I'm sending it in chat box. You can note it from there, please. Okay.
Also, Uzma, you can join as member of this society.
Yeah, exactly.
Okay, sir.
Already, I think one our friend is there, Dr. KK Gupta is from Ajmer.
Yes, sir. Dr. A.K. Gupta. Yes,
so
sir.
join this if you have resources and you can spare some money. Then you can join. Please join such societies which give you opportunity to link with nice people. This society, you can see lots of good people are there, serious and experts in their field. It will benefit you.
Yes,
Maybe
sir.
next time it will not member, you will not get the right information. That also talk to Dr. Patil.
Yes, okay, sir. Yeah, I'll be sharing all the details with her again. I hope she has noted my number now.
Okay, sir. You send your number in chat box. Okay,
Yeah,
I had taken a screenshot of this. Okay.
Yeah, thanks a lot. Thank you.
Okay. Thank you so much, sir.
Yeah. So let's close this meeting here. Very good night to all. We'll be meeting tomorrow afternoon again. Yeah. Thanks for joining. Thank you. Yeah. I'm concluding, sir, now.